All right, good morning, everyone. Thanks for coming to day 3. You all are the troopers who are here first thing in the morning, third day of a conference. Really glad to have you all here in person, and I know we've got a great audience tuning in virtually as well. I'm really excited that the one good thing that COVID brought us is it's made it just routine to, yeah, of course, live stream. So, if it took you a little while to finish breakfast or get the kids out the door, you can watch it virtually. So thank you all for joining. I am really glad that we do have a lot of people in person because it's great to make those connections that we have here at the IEP workshop. We're really excited to be in our beautiful headquarters building, but few logistical things again. If you want the agenda, there is an electronic copy available on the QR codes that are posted around. Bathrooms are out the door around to your right. The ones on the bottom floor, there's no key code. If you go upstairs, there's a key code that is posted on the walls of the auditorium. There is no food or drink in the auditorium besides water. Water should be in a water bottle or closed lid type deal, no open containers of water. If you're drinking out of a travel mug, I'm going to assume that's water. Please don't do anything to prove me wrong. And we do have coffee outside sponsored by ICF. So a huge thank you to ICF for sponsoring the coffee. We all know that science runs on caffeine. If anyone needs any kind of accommodation, help getting to the second floor, you do need a badge to use the elevators to get to the second floor. Listening devices, anything like that, please see Christine Job at the registration desk. The Wi-Fi is a CNRA guest, no password. As I mentioned, the workshop is hybrid. It's being live streamed, and most of the talks, those that have permission to record, are being recorded to post online later. That means that if you are asking questions, these will also be recorded, so don't say anything that you would not want preserved for posterity. A huge thank you to all of the organizing committee, especially Christine Job, who keeps being in the lobby while I'm thank her because she's working so hard. She doesn't actually get to see the conference that she worked so hard to put together. And Stephanie Fong back here doing all of the logistics and technical things. Speaking of logistics and technical things, if you are speaking today and have not uploaded your presentation, please do so at the break as soon as possible. If you had a poster upstairs in the poster hall over the past couple days, please pick it up before 2 o'clock this afternoon. That's when all the poster boards are being taken out of here and we'll have to hunt you down and find your poster. So yes, please pick those up. If you had any art that was on display, Christine Job has that at the registration desk. Thank you also to all of the volunteers who helped do timekeeping and mic running, and our California Natural Resources Volunteer Program who are running the registration desk outside, allowing all of the IEP folks who volunteer to do things inside so you can watch the presentations. And I think that's all of my boring announcements. So now we get on to the fun part. And first up, our plenary speaker today, I'm really excited to have Anke Mueller-Solger, who's currently the center director at the USGS California Water Sciences Center, former IEP lead scientist with a long career of working on science in the estuary and being a real heart of IEP, here to talk to us about the value of collaborative research and monitoring in IEP. So without further ado.
Great. Thank you so much, Rosemary, and thank you, Stephanie, and everybody else. Thanks for inviting me. Can't believe it's been 10 years since I gave my last IEP plenary talk. This is really true. And I strongly suspect that a lot of you just don't even know me at this point. And I'm really excited to see so many younger folks here in the audience. And I'm sure there's more online. Things have changed. Many things have stayed the same. Things have changed. This whole hybrid thing is obviously not something that happened 10 years ago, but there's advantages to it and disadvantages. Anyway, I have a somewhat odd title there. We are smarter than me, and it's also grammatically questionable. But I would like to tell you about working together and the value of that in the San Francisco estuary and of course beyond as well, and some thoughts that I've had about this over the years. And I will tell you a little bit about me, but before I do that, I will talk about we. Who do I mean with this we are smarter than me? And who I'm going to be talking about as we is we, the science people. And I do mean this quite broadly, but I will start with the IEP here as an example. So the IEP is an interagency program that was started by a group of people from actually at the time 4 different agencies coming together and deciding in 1970, quite a long time ago, right? Remarkably long time for a science program, to come together to create an ecological program. Actually, at the time, they called it Ecological Studies Program. It was the Interagency Ecological Studies Program. That S went away at some point. Really, it's over the years become so much more than just this 4-agency program. It's grown to 9 agencies, and it's opened, and it is open these days, to be a really productive, vibrant, open science cooperative and collaborative. There's been a lot of change and growth, including this venue thing here, or even the logo. I learned recently that changed colors because of ADA considerations, slightly. So thank you for pointing that out to me, Stephanie. And of course, many other things have changed as well. But what's never changed is that the goal of the IEP was to have people be able to come together, work together across agencies, across institutions, including academia, consultants, and so forth, to provide the best possible science for the management of the estuary. So that's never changed. And then I will tell you just a little bit about me, and I'll talk about me, but it could be any of you here. You're the science person. I'm Anke. I guess I'm not an interagency group. I'm more of an international traveler. I started that in Germany, and I've been in this country for quite a long time. I started in Germany in 1968, so I'm slightly older than the IEP. I came to California in 1990 and pretty much stayed after that. And then in 1998, I started working in the estuary. So Rosie was right. Oh my gosh, I've been around for a long time. I'm an aquatic ecologist with a PhD from UC Davis. My main drivers, the things that motivate me, are to help others do great work and to learn a lot of stuff along the way myself and help others learn. So that's what drives me. Of course, I've gone through plenty of change and growth of all sorts. Over time, one big thing happened in 2005, which is when I became a mother. And that actually did mess with me a little bit in terms of my identity. Me, the person, who am I? Me, the mother person, me, the science person. Can I combine the two? How is it going to work? Well, somehow it worked. And in 2008, I became the first ever IEP lead scientist, so Steve Culberson's predecessor. And I did that until 2014. And by the way, that's a hard job. It's a very exciting job, but Steve is doing hard work, and I want to acknowledge that because I know. And then in 2014, I switched to the USGS, where I was for quite a long time the Associate Director for Projects with the California Water Science Center, and now for a bit more than a year, I'm the director of that center, which sort of unfortunately, I have to say, means that while we do science for management, my own career path has been taking me into management, because that's what happens when you have a center of more than 300 people, 14 facilities, and all that sort of stuff. and crazy federal government bureaucracy on top of all that. So that's about me, but it's also about you. This could be any of you, and we are the science people. I thought a little bit about how we even got here, becoming we the science people. And it turns out that there's quite a bit of research about how Western science collaborations in particular, but just even this whole concept of science really got started. And you are, I'm sure, familiar with these two gentlemen behind me here, but they were quite instrumental and a big centerpieces of the scientific revolution that took place in the 16th, 17th century. And then there was also the Age of Enlightenment, and the whole thing there was that reason, science, reason, not spiritual revelation, would enable humans to make sense of the world around us and to gain knowledge, freedom, and happiness. That was a big deal, and it had a huge amount of societal consequences. So there were all sorts of revolutions that I'm sure you're all familiar with in the 18th and 19th century, the Industrial Revolution, the French Revolution, of course, the American Revolution, leading to we the people and thinking of ourselves and how we govern our states, et cetera, in a whole new way. And science was an important part of that. It's right from the beginning. But it also had a huge science impact because earlier there were basically a lot of individual, usually wealthy and mostly male, not only, but mostly male, folks who were calling themselves natural philosophers and all sorts of other interesting words. And they engaged mostly with science for science's sake, so basically basic science. And then this turned into a professional and merit-based, in the end, scientific community that conducted science for use by people, by society. And that really is where we, the science people, as we know ourselves today, came from. There are, interestingly, scientists who study how scientists work, and they've been doing interesting things to find out how this all studied. So I looked at some of these journal articles, and there is this one article where they mapped science collaborations in the 18th century, and that's what that's showing there. And they found, among other things, that really this started taking off in France, because France, with the French Revolution and the wars that were going on at that time, recognized that they had a need for scientific information to run the country. So they supported that, which made these collaborations easier. And then it spread pretty quickly around the world, including, of course, the United States eventually. And you can see that this continues to this day. So one piece of evidence for that, of people working together more and more. Science is not an individual sport anymore. It's a team sport. And you can see that by way of the number of authors co-authoring journal articles in all kinds of disciplines going up a lot. And that's the graph on the, I guess it's the left, with mathematics, chemistry, and physics shown here. But this is also true for other disciplines. So this is the average number of co-authors. It's going up there since 1960. And this is, of course, about the time of the IEP too, that the IEP started, 1970. And then the single authors are disappearing at a really pretty high rate. And in ecology, that's another journal article that showed that here. They just simply extended out this trend and found that by the middle of this century, there's going to be less than 0.1% of single authored ecology papers if the trend continues the way it is. So clearly we work together. We, the science people, work together. There are different ways of working together, and two really important words here are collaboration and cooperation. The IEP is cooperation, right? That's what's in the title. Cooperative ecological investigations in the San Francisco estuary since 1970. But as scientists, we're really used to this word collaboration. And one way to think about this, or at least the way I think about it, is sort of like an orchestra, using that as an analogy, where everybody is playing from one common score. And the important part is that there is a common goal, which is to play, maybe it's Mozart, maybe it's whatever, to play that as well as possible and really have the audience enjoy it. They've got a plan about how they're going to go about this. They've got the conductor who helps them get through this, and they've got resources, their instruments and the concert hall, etc. And then this ends when the collaboration ends, when the goal is met, when the piece by Mozart is done and everybody has clapped, and the resources are used up such that the lights in the concert hall go up. So this generally is how scientific collaboration operations work too. There's a project, we've got a clear goal, or we better have one at least, and we've got funding, and we've got all the materials, all the people, etc. And then by the time it's done, we might disband or perhaps start a new project. But that's a very typical thing that scientists do. But the other very important thing, and the IEP is truly all about this. In fact, I think, what did Steve say? Steve talked about how cooperation is, I think, the root of all the success for working across agencies, et cetera. Anyway, but it's a little different because you don't have to have the exact same goals. So all the IEP agencies have different missions, different goals, but they come together because the science thing is sort of an overarching need, and we are smarter than me, and I'll get to that too. It just really behooves us to work across these agencies, but the things are a little more independent, but we do, this is really important, have mutual considerations for each other. We need to care about each other and what each other's ideas are. Felicia Marcus talked about this a lot yesterday, how if you want to, talk with policymakers, you do have to understand where they're coming from, and sort of get into that mindset and consider their needs for real. And of course, they should also consider yours. But anyway, and there's going to be some mutual benefit. That's the whole idea. So then people, and I'm likening this to a drum circle, people can come and go in the drum circle. It'll just keep going, right? So it can keep going indefinitely. They don't have a score that they're playing from, but they're listening to each other, and they together make this amazing music. So there's this emergent property, to use an ecological term here, where the whole is greater than the sum of the parts that can come from this kind of thing. If we listen to each other, if we really take care of each other, that this whole thing really works out. So that's pretty simple analogies. But to put this into, the IEP loves conceptual models, I do too, to put this into a little bit of a conceptual model here. The thing about this is there are different parties here, and what we want in the end is that our science really gets used. That's why we do it, right? So there are some ingredients that are needed in order for our science to get used, and I'll get to that. And these parts of my conceptual models, they all sort of add up to the recipe for that. So the first thing I already talked about, it scientists collaborate a lot in all kinds of projects. Each one of you, I'm sure, is part of at least one collaboration, usually a whole bunch. That's what we do. And then in order to do that, we need some kind of, well, rules is perhaps too strong a word, but we need some kind of a structure. There usually is a project leader, right? There are lots of ways that we do things to make sure we do them right. The IEP and pretty much any science organization out there has a review process. That's a really important thing. The USGS has this, and I really like this about the USGS, by the way. It was one of the reasons why I wanted to work for the USGS, has this strong set of guidance and rules called fundamental science practices that are there to, protect scientific integrity and quality of the science done by the USGS and the scientists who do it. So those kinds of things become pretty important. I listened too much to Rosie. I kept it close. Sorry. All right. So again, this is super important that we have these rules and that we are clear about what we're working on together, et cetera, to keep our science credible, which is actually the very first ingredient needed in order to be able to get our science used. If people don't think that you did it right, and if you yourself don't even believe it because you guys didn't follow your own rules or whatever, it's not going to be credible. Of course, scientists also cooperate a lot. So in this particular workshop here, this isn't one project, but there is work going on. When you talk with each other, when you give each other ideas, when you give each other feedback, that is actually work. And that's basically this looser thing that can come and go, and there aren't all these rules, etc. But it's super important. The next group that is part of this, and by the way, they're also part of we, the science people, because they use the science, are the resource managers and the policymakers. So Felicia Marcus again talked about that a lot yesterday, and I'm certainly not going to repeat her talk, but it's super important that we, as scientists, communicate with research managers and policymakers. And I suspect there are some perhaps in this room here that would consider themselves be in that category too. So we cooperate with them, and they, of course, also cooperate with each other. For example, the IEP directors are generally not scientists, but they are working together cooperatively in the interagency ecological program. Because they realize that this is where good science comes from, and this is a good way of doing this. And they talk about that a lot. Okay, and then this is super important because this is who uses the science, research managers and policymakers. So in order for our science to be relevant, which is the second ingredient here, it's very important that we talk with these folks. The IEP is obviously cooperative investigations. That's what it's called for a reason, because this cooperation is so fundamental and important. I like about the USGS that more than 100 years ago, folks recognized that this is a good idea across agencies. So actually even Congress recognized it and gives money to the USGS to match money that the USGS might get from others or that others have to really work together. USGS cooperative matching funds have been around for more than 100 years because cooperation is so important. And then the final group is, of course, all sorts of other stakeholders, community members, NGOs, whoever, who has an interest in what's going on in, say, the San Francisco estuary. They might co-develop some of the scientific projects that we do. They might, in some cases, co-manage some resources. And they will watch what's going on. And it's so important that we are quite transparent about what we do and that we keep talking with these people, because the third ingredient here of making science usable and get it used is that it's also got to be viewed as legitimate. Was there a reason why you did it and not someone else? Was it done right? Did we ask the community how this might affect them when it comes out? Felicia Markets mentioned one really good example yesterday of the San Francisco Bay Shoreline Adaptation Atlas. It's a long word, I had to actually remind myself, but which was something that was produced in the San Francisco Bay area and where the lead authors talked a lot with community members before the thing ever came out. So then when it did come out, the community members embraced it. Otherwise, they might have been potentially even, struck as this was something that they wouldn't want. But they knew about it. It wasn't news to them. They actually contributed a bit. And so this was a product that now the community can embrace and use. So that's really important too. But in all of that, obviously, super important that we simply keep talking with each other, that we keep working together. Steve Culberson was talking about the constant dialogue that we need to have in order to have successful cooperation and success through cooperation. So sometimes that's really hard, right? So I want to acknowledge that. And why is it hard? And I know there's some hard times also in the IEP. This is no surprise because there's a lot of people here, and human relationships, that's thing one, are just tricky. We're humans, we all come from different places. It's hard to work together at times. For scientists, especially folks in academia that are trying to get tenure, or in the USGS, we have this research grade evaluation thing where individual contributions are being evaluated. This can be tricky sometimes because it might be hard to pick out individual contributions from the group. There are differences in motivations, goals, culture, etc. So that also comes into play a lot when you try to work in groups. There are risks with forming groups. There can be groupthink. There can be exclusion of other people who have different ideas or different values or whatever it might be. It can become terribly rigid as soon as you organize something. There can be internal conflict. You can be more exposed to external pressure because you're more visible as a group. And this actually happened a lot in my day. In the early 2000s, when we came upon the pelagic organism decline, as we then called it, in the Delta, and I'm sure you've all heard about it, the pod. It's on the bingo cards, I noticed. Where then IEP scientists had to dress up in strange outfits and do strange things. And there was a lot of pressure, believe me, to get through that. There is a need for support, especially when times become hard. Support from other people. It doesn't work without support. So again, in France, this whole collaboration thing really started because there, as opposed to elsewhere, because the government actually supported that. There's a need for, and I already mentioned it, internal organization, coordination, also conflict resolution at times. There can be increasing bureaucracy and death by meeting. I suppose all of you have been there and at one point or another. And all of it can then add up into individual and even group stress, exhaustion, frustration, lots of bad things. So why the heck do we do it anyway, right? One would wonder, well, because it's smart, right? We are smarter than me. There is simply no two ways around that. There is a lot of intellectual enrichment or cross-fertilization and all that kind of thing that I hope you all have actually experienced at this workshop here the last few days and will still experience today. Things you would never think of if left to your own devices in your own room or something. So super important for science to make progress. Of course, it then also gives you access to diverse knowledge, skills, lots of equipment that's being shared across the IEP, facilities even, or hearing about these really important scientific needs or questions that resource managers, policymakers, or even other scientists have that you might have never thought of yourself. Which then adds up into overall more efficient and effective use of time, labor, and equipment, and greater productivity with less, which really the folks who are supporting these sorts of things should appreciate and perhaps understand even better than they do. But that's how we make progress, especially when it comes to tackling the super complex problems that we have here in the San Francisco estuary. Yes, they are extremely complex and have an impact, make a difference in helping resource managers dealing with some of these problems and have all of this then be relevant to them. Another thing is quality assurance, quality control. It is better when we look at each other's stuff and keep each other on the straight and narrow, right? So the IEP has a lot of review processes. In fact, this is how I got my start in the IEP, is when I was hired by the Department of Water Resources in 2000 and engaged or was there to co-lead with Zach Hymanson, some of you might remember the name, a review of the environmental monitoring program. It was a pretty sort of large-scale review that resulted in lots of things, including, and the younger ones of you probably don't know this, including a change in the name of the program, because it was called the D1485 Compliance Monitoring Program. Doesn't exactly roll off the tongue, right? Right, I didn't think so either. Anyway, so talking together, we realized, that was a relatively minor thing, but it was a thing, and it is there, it has made a difference to this day. But more importantly, the quality and what we really do there was, of course, what we looked at, and that was really important. And the IEP continues to do this. The Delta Science program does this a lot. Every university does this. I can't be at this here workshop very much this week because we have internal center science reviews, project reviews going on right now that I have to be at. So it's very important, and we do this together. We can't do this alone. And then finally, overall increased visibility and recognition. People know that the IEP puts out great work. Well, some people know that more than others. But anyway, which gives it, a great amount of legitimacy. I can't say that right now. Legitimacy. I think I even, whatever. And then finally, like that picture right there, I felt very connected to these people. I felt like I found a group that I belonged to when I first started with the IEP. I hope you feel that way about your colleagues, about each other here. But this is something that's not to be underestimated. And also, it really counteracts some of these negative things like the death by meeting and the frustration when you have, some conflict with somebody, etc. This is really an important thing too. All right, so we, the IEP science people, work together, and what do? Of course, we do a lot of monitoring, we do a lot of research. That often comes together in the form of adaptive management, which is a thing that's been around since 1978, formally at least. The Delta Plan adopted it in 2013 as a way to bring the science and the management close together. The IEP did it before the word actually existed, or this thing as a concept existed. And for example, here, this early paper by Jim Arthur and Doug Ball, which used a summer-fall of outflow action to study the significance of the entrapment zone, as they call it, on phytoplankton. And of course, that's something that spawned enormous amounts of additional research over time, along with regulations like X2 and things like that, and ways to manage the system, and then had successors, such as, for example, in 2011, when there was a fall outflow action, and then the IEP, and it was 17 authors in that case, wrote that up. So by the way, 2 authors to 17 authors. So we work together more and more, yes. And then most recently, very recently, just came out this amazing special issue here of the San Francisco Estuary and Watershed Science Journal, led by Rosemary Hartman over there, the IEP synthesis group working on this, about drought impacts on the estuary, which I think is just really remarkable. So whoever was all involved with this, or even if it's, just collecting data or somewhere on the sidelines, congratulations to that kind of work. So we, the IEP science people, work together on new IEP and science frontiers too. And I have noticed on the agenda here that there are quite a number of talks about bringing in the human dimension of what we're doing here more and more, social science, etc. So bringing science and society closer together than they have been in the recent past, at least. So we, the science people, and we, the people, yeah, and I did notice that there were several amazing young people, thank you for your work, who are working on exactly these types of things. And I find this very exciting. I think we'll see a lot more of this. I'm wondering, and that's just me, and it's perhaps a little out there, if perhaps at some point there'll even be some spirituality brought back in, because during the scientific revolution, that was basically tossed. Never mind that the spiritual stuff. But we do need to be inspired to do our work, and we are inspired, right? And somehow, what might that mean? We don't know. I have no clue. But some years ago, who would have thought that we would have talks such as interweaving traditional knowledge and Western science at the IEP Science Conference? That was totally not something that people thought of when I started with the IEP. So I'm so glad to see that. So we, the IEP science people, work together. We talk shop. That's why this is called a workshop, folks. It's not called a conference, because this is about working. It's always been that way. It's traditional. There's a lot of work going on here. And I really like this about the IEP. I've always loved this. People really work with each other, talk shop, have a workshop, have all the project work teams, which are just, in my opinion, basically the best thing about the IEP, where people can come together and cooperate and sometimes collaborate to really produce this incredible science that you all are producing. So great science, great people. Thank you, IEP science people, and thank you again for having me at this workshop. It's lovely to be back 10 years later. Okay, thanks.
Thank you so much, Anke. Does anyone have any questions for Anke about any of the collaborative science topics that she brought up here?
No Delta Smelt in it, sorry. No Longfin Smelt either. Yeah, it's actually kind of weird for me to do a talk like this. It's still weird, without lots of science graphs and things like that. But it really is worth thinking about these bigger things, about working together. And the IEP is just such a model for how this can be done and persist. And, the IEP's gone through lots of ups and downs, and I think there's some of that going on right now, but it's persisted. And the main reason is it's smart. We are smarter than me. It really makes sense. Cooperation is the reason for success across agencies, et cetera, doing science together. That's what we do. So thanks again for having me.
Thank you so much. And there's a couple graphs in there. Now I want to chart number of authors on Steve's master list of IEP publications, see how that relates to time. All right, well, now up, we have our next regular old science talk full of lots of graphs. And for the next session, we have Vanessa Tobias, who's going to be introducing the speakers. Yeah,
We can do it quickly. So, okay, so we're going to start off the Longfin session. And let's see, so we've got, first up, we've got Nikolas talking about maturation and fecundity. I'm really excited about this.
Good morning, everyone. Okay, so thanks for coming up for early talks of day 3. So since I'm kicking off the Longfin talk, well, first I'd like to start by acknowledging our funders and a bunch of my co-authors. So I'm Nico Floros. I work in Levi Lewis's OGFL lab at UC Davis. We are doing a big Prop 1 funded project right now with a ton of people. So thank you for everyone. You know who you are. I wanted to start with a little bit of background on Longfin for the folks in the crowd who don't know a ton. They're semi-anadromous. They range from all the way up in Alaska down to here in the San Francisco Bay. This is the southern extent of their range. And recently we figured out that the Longfin that live here in San Francisco Bay are genetically distinct and are contributors genetically to the rest of the population up the coast. As I'm sure almost everyone here knows, they have been in stark decline, like many species here in the estuary. They were listed as threatened on the CSA in 2009, and in 2024, they will very likely finally be listed as endangered federally, which means that we should really be thinking about how we manage these fish. To do that well, we need life history models, and we need population models, and we need those to be informed by science. And so, cue the big Prop 1 study that is going to try to answer a bunch of these questions. So I'm sort of one half of this big project, and so I'm just going to talk about sort of my half of the thing. So the objectives for us on the fecundity maturation side was to figure out, do Longfin smelt synchronously develop all their eggs? So do they develop their whole gonad all at once? And then figure out how to stage those gonads, get maturation staging for the gonads, so we can categorize that and then talk about the overall fecundity of fish. So to do this, we had to get our hands on a bunch of fish. At the OGFL, we do our own collections, mostly down in Alviso Marsh in the south of the bay. But we also got a ton of help getting fish from folks at CDFW. I want to particularly call out Christina Birdie and Trisha Bippis, who were a huge help in helping us comb through CDFW's massive archive of Longfin smelt that is huge. So at the end of all of that work, we had over 1,600 fish for which we could do maturation staging on. The 1,600 fish span over 20 years. They're all over the bay, and they vary in almost every month of the year, but of course, primarily in the spawning season in the winter, as you can see. So once we had all those fish, we had to get things that we needed out of them. So we dissect them, we get morphometrics, we remove the ovaries from their body cavity along with a bunch of other tissues and also their otoliths. We take those ovaries and we stage them. So I'm going to walk through this staging rubric in case there's folks who don't know this. A lot of people maybe are familiar, but basically it's a 0 through 6 scale. 0 is there's no gonad. 1 through 3 are essentially immature developing eggs. 4, 5, and 6 are all sort of the big distinct different stages. 4 is a ripe, ready to spawn female. 5 is an already spent female. There's no more eggs left, or there's almost no more eggs left. And then 6 is this weird, unusual thing that we've been seeing as we go through this, which is what we think is evidence of batch spawning, so multiple clutches in a season. And we can, well, in fact, I'll show you in a second in a picture how we know what that looks like. So here are sort of just 3 of those 6 stages to give you some context. So stage 1, super young, super early, immature gonad. The oocytes are very small. They kind of just look like a cloud unless you look at them under a microscope. Stage 4, you have these big, ripe eggs. They look like little fish eggs, just like you might see on sushi. And then stage 6 can look a lot of different ways. This one's a really good example because you can see in and amongst those sort of stage 3-ish eggs, there are 3 or 4 big, old, we think, sort of atrophied dark yellow eggs. that we believe were left over from the previous batch of eggs. So here's a nice little photo of those two lobes of the gonad. With Longfin, the left lobe is always much larger than the right lobe. So here's the left and the right lobe of the Longfin smelt. To do our fecundity analysis, we essentially took 3 spatially distinct sections out of the left lobe, and we can use each one of those we can count all the eggs in each section and get an estimate that we then can say for the whole lobe. And then with the right lobe, we would just count the entire lobe, or if it was quite large, we would like break it into 2 sections to count it. So with each one of those sections, we take it out, we remove the eggs from all of the sort of gonadal tissue that's not the eggs, and we can flatten those eggs out into this nice, pretty flat plane, and we can photograph it. And once we photographed it, we can use a program, a plugin for ImageJ to go through and digitally count, which is fantastic. It allows us to A, not destroy a lot of the gonad, and B, have something that we can go back on and do QC on. And again, I want to point out that we are taking these 3 spatially distinct sections from the left lobe. So first off, we get 4 separate estimations that we can use for the fecundity of the fish. And also on the left lobe, we have these spatially distinct estimates, so we can look at if something's going on at one side of the lobe that's not in the back. So maybe are the eggs more developed in sort of in the anterior, the posterior, or are they all developing simultaneously? So results, do the eggs develop synchronously? So here I'm showing a figure of egg density relative to those 3 sections. And what we see is egg density is a good proxy for maturation, by the way, because the eggs get bigger as they mature, and so they get less dense. And egg density is the same across a single lobe. If we compare the left and right lobe, egg density is the same between lobes. So we have pretty strong evidence that the eggs are maturing all at once. If we go into staging, I want to take a second to orient you to this big figure because I'm going to show a couple of them. So colors are stage. The sort of gradient of blues are those 1, 2, 3 maturing eggs. Bright orange is a ripe female ready to spawn. Red, she's just spawned out. And then black are those stage 6s. Maybe she's getting ready to spawn a second time. So relative to size of the fish, we see, there we go. Basically, they start maturing around 70 millimeters. That's where we see most of the fish are like really getting beyond stage 1, so starting to mature. By 90 millimeters, most of the fish that we see are either ripe, ready to spawn, or have already spawned out. And you can also see that those stage 6s sort of increase slowly as you get bigger and bigger fish. I'll point out, in case anyone has eyed the 120, that looks very strange. If you look at the total count, you'll see that it's because there's only like 3 or 4 fish that are that big. So if we look at the same thing, but now we're looking across the year seasonally, as you might have anticipated, they mature during their spawning season. That maturation really kicks off around November. And as you see, you get more and more ripe females, as more and more ripe or spent females as you move through the spawning season. Now for the big stuff. Fecundity. So here is total estimated eggs compared to the fork length of the fish. It goes up. There's not a. It goes up with fork length. There's a little bit of an allometric curve, but it seems kind of linear. So we wanted to put this in historical context, see if these results look different than some fecundity metrics we have from the past. So here's those exact same data, just simplified down to compare to some other studies. And I want to point out real quick, I don't think I have a pointer. No, I don't. But you can see there's one fish over there that looks like, oh, he probably should have removed that. That's clearly an outlier. It's way more fecund than any other fish. So just keep an eye on that one. So these are all our data. For the fish that we've done fecundity on so far, we're in the sort of like early 2000s all the way up to 2022, I think is the most recent. And if we compare that to data from Randy Baxter back in the 1980s, 1990s, we see that our fecundity matches his pretty closely at small sizes, but the fish back in the 80s seem to be significantly more fecund once they get past that sort of 110, 120 millimeter length. And our outlier is not looking so much like an outlier anymore. And now if we compare it to Chigbu et al. Study from way back in the 1960s, we see that they were a lot more fecund back then. So just, I want to highlight the difference there. So if we took at a fish that, let's say, is 120 millimeters, one of the fish that we see at 120 millimeters, we would expect to have around 7,000 eggs. Back in the 80s, we might have expected 10,000 eggs. Back in the 60s, would have been essentially double what we see today at 15,000. And I want to remind everyone that we're pretty sure we're seeing batch fecundity. So I'm not giving you the total fecundity of the fish for a season. I'm giving you one batch's fecundity. So when I say that back in the 60s, they might have had double the eggs, that's per batch. So maybe in a season, they would have put out 1,400 eggs today. A long time ago, they might have put out 30,000 eggs. So we're really seeing a stark decrease. We think, at least, we're seeing a stark decrease in fecundity for those larger size fish. So just to recap all of those takeaways, we seem to think that they synchronously develop their eggs. That maturation of eggs seems to happen starting around 70 millimeters, and by 90, most of them are developed. And the average fecundity has reduced compared to the past, especially for larger fish. And I want to reiterate that this is sort of one half of a big project. As I mentioned, when we're dissecting those fish, we're taking out those otoliths. I work for Dr. Levi Lewis, who, if any of you know, is an otolith guy. I happen to be the guy doing the not otolith stuff in the lab. So I just wanted to call out the poster by Alex Lama, which you have a little bit of time left to see today. If you go up and maybe you can find him walking around. He's essentially presenting on the other half of this big project, which is all of the otolith stuff. So we're taking the otoliths from the same fish that I have these gonads for. We're aging those otoliths and trying to tease apart not just size differences in fecundity, but also age-based differences in fecundity. Yeah, and so with all of that, I guess I'll take questions. Folks are coming with microphones.
Thank you. That was great. I was wondering, is our Plenary Chigbu looked at a lot of fish in Lake Washington?
Yeah.
Is that where those are from, or are they from the?
So those are the Lake Washington fish, which
Okay.
which
are genetically distinct.
Genetically distinct, completely life history distinct, right? They live in a lake. They live farther north in the range. There's a bunch of distinctions between them, but it is. The reason I put it in there because these fish could do this given the circumstance, right? Like
Maybe.
who knows?
Okay. All right. Thank
Yeah.
So my question is about what you think is the cause of this. So potentially genetics, or did you see any association with condition and fecundity?
You said the cause of this. You mean?
Oh, sorry. The dramatic decline in
Sure.
fecundity.
So that's sort of the third leg of this project, is looking at sort of spatially across the bay and across those years, looking at how condition changes, how hepatosomatic index changes, and all of that. We're still super working on this data, so I don't want to try to draw conclusions from it. I think there could be a larger scale, simpler answer to this, which is that we are at the southern end of the range of this fish, and climate change is causing ranges to shift north. So we are probably approaching, we're probably making like more and more difficult for this fish as their range would naturally shift farther north with climate change. That could be what's going on. Yeah.
So you're thinking temperature, not necessarily food deprivation.
I mean, well, it could also super be food, right? There's been a huge change in the food webs in the last, 100 years in the estuary. Yeah.
Cool. Thank you.
Anyone else? Sean again.
All right. So if you're asking about the food, have you been able to examine? I know you don't have very many of them, but what about the cultured fish? The fecundity of those?
No, we haven't. We have some to process, but not very many, and we haven't done them yet. But that's on the block to do. Yeah.
Yeah. Then you can test the food theory. Okay. Thanks.
Yeah. We also, I will, a little shout out. We take the otoliths, we take the gonads, but we also take gut tracts. So we haven't touched anything about the gut tracts yet, but if someone's looking for diet study stuff. Thank you, everyone.
All right, so next up, we've got Morgan. And Morgan's going to tell us about the larval entrainment study.
Thank you. I hope you all are as caffeinated as I am this morning. Though if it ends up seeming like I'm speed running this, we all know we can blame that ICF coffee. The other reason I might seem like I'm talking fast is that I've got a lot to talk to you about today. But the core of it is that less is more. In this case, it's more data, more replicates, and more answers that modelers can generate for managers. I'm going to be telling you about the larval entrainment study, which is a new companion study running alongside the Smelt larval and 20 millimeter survey programs. I'll fill you in on the logic behind this study, and then run you through a short history of our sampling, including summaries of some of the special studies that we've completed this year, and then wrap up with a little bit about what we're planning on doing into the future. Before I get to all of that, I want to take a moment to thank our funding agency, the Department of Water Resources, and also acknowledge the many collaborators that have participated in developing this project, including the staff of the CDFW Native Fish Unit and Fish Facilities Unit, Water Branch, the Department of Water Resources, and the U.S. Fish and Wildlife Service, and of course the many people that have participated either in the field or in developing the design of this project. We could not do this without you. Anyhow, let me loop all the way back to the beginning. As Vanessa mentioned, my name is Morgan Gilbert. I'm an environmental scientist working for the California Department of Fish and Wildlife, and I work for the Fish Facilities and Entrainment Unit. You could even call me the E in FFE. And that's because I lead the larval entrainment study. We started that study as the Larval Entrainment Pilot Study, or LEPS, back in 2022. And at some point in the last year, we renamed it to just LESS, which transformed me from a leper to a lesser. I'm still not sure whether that's an upgrade or not. But the P there stood for pilot, and we're continuing in that vein moving forward. By taking an adaptive approach toward developing our study and addressing questions about the entrainment of longfin smelt. Entrainment's an important issue, both from a scientific and a policy perspective, enough so that the incidental take permit for the long-term operation of the State Water Project includes Condition of Agreement 7.6.2, which calls for a study to quantify longfin and Delta Smelt entrainment into the Clifton Court forebay, which I've circled here in red, down in the southern reaches of the Delta. Entrainment occurs when fishes are diverted by flows out of their preferred habitats and into neighboring regions. A lot of the time when we talk about entrainment, what we're thinking about is direct entrainment, right? Which is fish being drawn by flows into the water facilities. But that's actually just part of the problem. It is an issue when fish are brought in directly, they become isolated, they experience mortality through a variety of mechanisms, but also present is indirect entrainment. And that occurs over a broader area. And that happens when flows carry fish out of their preferred habitats into areas that might be lower quality for a variety of reasons. They could be stressful, dangerous, and that tends to have negative consequences for the individuals and populations where that occurs. And so a particular concern for our study is larval entrainment, which among other things can negatively affect the population's ability to recruit new members. We know that some direct larval entrainment is happening, and that's because of the qualitative larval sampling program that the facilities runs. Unfortunately, salvage is centered on diverting fish from the facility and not necessarily in quantifying the take of those fishes. And because of that, the methods that they use make it difficult for them to quantify the number of fish that are being entrained. There's low retention of larvae in their sampling. And so while salvage is a crucial step in mitigating the entrainment of adult fishes, it really can't tell us very much about what is happening at the facilities with entrainment, other than that osmerids are present and are being entrained. And so when we start to address this problem, we find ourselves joining a conversation that's been going since the 1960s. back when the facilities were first constructed and biologists were starting to consider the impacts of entrainment. In fact, the osmerids I'm talking about today were far from the first fish that came to the attention of biologists and managers with impacts from entrainment. This 1985 paper from Stevens et al. Found that their data suggested that the entrainment of striped bass larvae was likely contributing to their decline in the system. The first alarm bells regarding the potential impacts on Delta Smelt came in 1992 from this paper by Mueller et al. The authors here suggested that the entrainment of larval and adult smelt could be contributing to that species decline. And then the federal listing under the Endangered Species Act of that species in 1993 had pretty profound implications for the operation of water extraction facilities within the Delta. That's prompted a long scientific conversation about longfin and Delta smelt and their entrainment. In this 2021 paper, Smith et al. Describes their modeling effort to explore the role that entrainment, among several other factors, plays as a driver of Delta smelt mortality, particularly during dry years with turbid conditions. And in their conclusion, the authors note that fully mitigating losses to entrainment is unlikely because human needs mean that water exports and entrainment related mortality will be very difficult to reduce to zero for these populations. According to the authors, managers will need to accept some degree of attrition. Other modeling efforts have come to different conclusions about the impact of entrainment on longfin smelt. In their 2022 paper, Kimmerer and Gross described a particle tracking model that showed that under a variety of different hydrological conditions, that passive particles that represented larval longfin smelt were unlikely to be entrained in large numbers. And that's because their simulation showed that model smelt released in inferred hatching regions would drift seaward until either they were large enough to control their own movement or were just swept out to sea. Alongside further analysis, they concluded here that losses of longfin to entrainment are likely to be small in magnitude and that other factors will be playing a larger role in the species population dynamics. And so, that's why I'm here. We know that entrainment could be affecting the population dynamics of longfin, but we don't necessarily have all the answers that we need to mitigate those impacts. So less exists to address this gap and give managers a better picture of the cost of pumping while providing modelers with ground truthing and more data for future analysis. And now I get to tell you about what we've been doing out on the water. We started this project in 2022. That year, our sampling was conducted at West Canal, right adjacent to Clifton Court Forebay. We sampled 5 days a week. We started using the same methods as the smelt larval survey. So it's a 500 micron mesh net mounted to a small sled. We did oblique tows where we sampled the whole water column. That ran from January to the middle of April when we switched to a 20 millimeter survey net, which is below there. It's a larger sled with a larger mesh, 1600 micron, and that wrapped up in mid-May. We collected 41 longfin between January and March and saw a pretty steady increase in overall fork lengths until we switched to the 20 millimeter gear. And around that time, our catch of longfin dropped to 0. That densities of longfin had peaked in February and were declining by the time that we made that switch. So that lack of catch likely just indicated that larval longfin were no longer present in the area, but it nonetheless sparked an interest in alternative gear types coming up in our 2023 season. Now, I've actually got some footage here of my very first day, January 3rd, 2023, of me coming on board the larval entrainment project. And as you can see, I've gotten a lot taller. But my personal growth aside, I was not the only newcomer this year. We also added a new net, which was a 940 micron mesh that we mounted to an SLS sled. Our sampling this year alternated between the two gears, the 500 and the 940 nets, which made our season something like one big gear comparison. We discontinued our 20 millimeter sampling due to the low catch that we observed in 2022 and sampled actively from January through April. Likely due to the relatively wet hydrological conditions that we experienced in 2023, we caught relatively few longfin smelt this year. The species appeared early in our 500 micron sampling and later in our 940 micron sampling. Across all taxa, we caught a variety of lengths with a mean around 7 millimeters. Our catch was mostly dominated by the native prickly sculpin. Our new 940 net did appear to be more efficient in capturing larvae over 6 millimeters, but overall our 500 micron net caught the most fish overall. In 2024, we continued our adaptive approach by expanding out of West Canal to a series of SLS stations located along the Old and Middle River corridor. I've got two marked out here at the top of the OMR corridor, which I'll talk about in a second. But I want to mention that while our sampling in West Canal was doing a good job at detecting longfin and their direct entrainment, as we saw, that represents only part of the effect that pumping can have on their distribution. And so these two stations in red that I just called out, those at the top of the OMR corridor were our priority or early warning sites, which received intensive sampling on the Monday of each week. And then on the remaining sampling days, we ran a longer transect, starting at those stations and then running down kind of the whole OMR corridor. And our hope there is that that will generate a broader and more consistent picture of what longfin distribution looks like within the zone of entrainment. Now, in 2024, we had hoped to continue sampling with our 500 and 940 micron net side by side, but I got to tell you folks, the sea can be a harsh mistress. Our 940 net was lost during sampling very early in our season. It was nicknamed Captain America, and so maybe we'll be lucky and he'll make a triumphant return sometime soon. But without him, we moved on and turned to the new hero, which was the Wonder Twins. In that case, they took the form of a bongo sled, which is named for the drums that they resemble, and that I am very merciful in not using as the soundtrack to this talk. Instead, please just enjoy a picture. We undertook oblique tows with this gear, which allowed us to sample a higher volume of water with the same number of tows, hopefully providing us with a better chance of detecting the rare species that we're looking for and improving our ability to assess their densities at those sites. Developing this gear was a major investment during our 2024 season, including extensive work designing, fabricating, and testing it. Our first gear naturally broke immediately, but it taught us a lot of important lessons. With additional reinforcement and the addition of a sled and frame, we found that this gear sampled very similarly to an SLS sled in terms of its performance in the water and its depth profile. And this initial testing was so successful that we ultimately switched to this gear exclusively after just the first few weeks of our sampling season. We're very excited to share our 2024 results. We're in the midst of processing this season's data, so please stay tuned. Here, I'm going to give you a quick whirlwind tour of the special studies that we performed these last few years. In 2022, we carried out a series of day-night comparison studies in January, March, and February. Got those kind of mixed up. This study is actually written up in detail in our forthcoming IEP newsletter article. So if you want more details, please look there. But in summary, we found that our early morning and evening sampling encountered more longfin than our daytime sampling. In 2023, we set out to more closely examine the three gear types that we have been using between the 500, 940, and 1600 micron. We used 3 SLS sleds mounted with those nets and found that we were sampling too late in the season, unfortunately, to reliably catch longfin. But across all species, we found that the 500 continued to have the highest catch overall. But we did find that the 940 gear was somewhat more adept at capturing larger larvae, particularly of some of the more common species like striped bass. Our analysis of the data showed that the 940 and 1600 micron nets produced pretty similar results, and the 500 had a distinct advantage in overall catch. We also did some night sampling. I'm going to speed through this because we don't have any results yet, but I can't really undersell how pretty it is to be out in the Delta at night. Unfortunately, I don't have any good pictures to show that. So all you get is this picture of one of our samples and then kind of this horror movie version of the bongo coming up out of the water. Call it the dark bongo. But please stay tuned for that. Those special collections are being processed along our 2024 samples, and we're excited to share those results once they're in. As we look ahead to 2025, we're planning on continuing our core sampling within the high-risk OMR corridor. We're also planning to pursue additional special studies like surface bottom comparisons that will allow us to address the effect that our oblique toe methods have on our net's efficiency. And we also plan to carry out additional gear comparisons to critically examine the effectiveness of our equipment. I'd like to highlight that we're planning on publishing our data on the Environmental Data Initiative, making it freely accessible to all of you in the greater scientific community within the Delta. And then I'd like to close out by noting that we're collaborating with DWR's Genetic Monitoring Lab, and we hope to be able to provide them with some ground truthing as they work on developing an eDNA assay that can detect the presence of longfin smelt. Stay tuned for more information. And if I can just wrap up real briefly, I'll just tell you that's the story of how less became more. We're still growing. I hope that we can advance the conversation around entrainment, and I'll close by saying that part of achieving that goal is engaging with broader IEP community. So you're welcome to catch me during the break because I'm pretty easy to find. And I think I'm out of time, so I won't take any questions, but thank you all very much.
Thanks, Morgan. Sorry, we don't have time for questions, but like Morgan said, that's what the breaks are for. So now we are going to start the Vanessa's talking about longfin section of the longfin group. So this, we're gonna have Vanessa Mora talking next about the expansion into San Pablo Bay.
All right. Hello, everyone. My name is Vanessa Mora, and I am an environmental scientist within the Native Fishes Unit at CDFW. I am one of the project leads for the Smelt Larva Survey and the 20mm survey. I'm here today to talk about our expansion into San Pablo Bay. All right. The Smelt Larva Survey was initiated in 2009 to document the abundance and distribution of larval longfin smelt in the San Francisco Bay estuary. Historically, the survey conducted bi-weekly sampling from January through mid-March among 44 fixed stations. The 20mm survey was implemented in 1995 in response to the federal ESA listing of Delta Smelt as threatened. It runs every other week from mid-March to early July for a total of 9 sampling weeks. This survey has historically sampled 47 fixed stations. While 20mm targets post-larval to juvenile Delta Smelt, it is also useful for monitoring early juvenile longfin smelt. Both projects are used by agency managers to assess the vulnerability of listed osmerids to entrainment by the South Delta export pumps. Both conduct 10-minute oblique tows and collect samples that are preserved in 10% formalin for later identification. A couple of differences to note between the two projects is that SLS is most effective in capturing 5 to 15mm larvae, while 20mm survey averages a wider length frequency from 10mm and up. The SLS gear is the top left picture, and although it looks similar to the bottom right picture of 20mm gear, it is very different because of its smaller sled frame and mesh size. SLS does 1 tow at each station, whereas 20mm does 3 replicate tows, including a concurrent zooplankton tow. These projects are well established, but the spatial and temporal distribution of larval longfin smelt was found to extend beyond the historic sampling frame for both projects, which limits the project's representation of interannual variability. In response, we took steps to help improve representation. In the 2022 sampling season, we added 2 supplemental surveys in December to SLS to help us detect larvae sooner than the first sampling week in January, when we usually already see longfin smelt catch. Starting in the 2023 sampling season, we increased the sampling effort in San Pablo Bay, where larval and early juvenile populations reside during wetter years. All right. The monitoring survey design team is a combined effort of multiple agencies with several goals, but I'll only be focusing on the ones relevant to 20mm and SLS. One of the goals of this effort is to implement design-based estimators of abundance to provide a more standardized method for estimating abundance across projects and species. Another goal is to improve spatial balance by strata within and between projects. to increase certainty and improve species detection for the abundance estimate. So these two goals go hand in hand. The picture on the right shows different regions, strata, and subregions. One of the ways to improve spatial balance is to increase the number of stations in areas where population estimates are desired. For 20 millimeter and SLS, Arun Malwani's analysis indicated the priority region for increased sampling is San Francisco Bay, where the abundance estimate standard error could be reduced by 25%, with 6 to 15 additional toes per sampling week in the region. This is a map of where the SLS stations fall into the different regions and subregions. Our focus will be San Pablo Bay and Carquinez Strait. All of these new stations are actually pre-existing sites for other surveys, except for 405, which is already a routinely sampled station for both projects. 323 is a routine 20 millimeter station. 328, 335, 329, and 336 are 20 millimeter high outflow stations, and the rest are current fall mid-water stations. There were certain considerations during the selection process, such as including a mix of deep channel and shallow shoal stations, and making sure each station was at least 2 kilometers apart. And then very quickly, this is just a 20 millimeter map with all of our stations. A lot of the stations are the same, except for some areas like the Napa, we have less stations, and we have more in the North Delta. For the San Pablo expansion, though, they're the same stations that we picked for SLS. The reasons for the move to design-based abundance estimates are: it proved flexible and efficient for calculating project-specific regional estimates of relative abundance, along with the associated uncertainties. It provided a standardized method for estimating abundance across the studies and species, and it can be applied in fixed or probabilistic spatial designs. Essentially, the design-based abundance estimate is CPUE times volume, where the CPUE is averaged across stations within a region and then multiplied by a regional water volume. The real key is that this approach allows for variance calculations. The graph depicts the design-based abundance estimate for Longfin smelt and SLS, and the gray is the confidence intervals. The well-established population estimation approach employed in this effort is adapted from Polanski et al. 2019. All right, SLS added effort. We added 15 stations to the San Pablo region, bringing the total number of stations for a sampling week from 44 to 59. It added a full extra day downstream. Overall, it adds 120 samples to our sampling year for SLS. The stations highlighted in yellow are the additional stations. For 20 millimeter, we added 14 stations to the San Pablo region, bringing the total number of stations per sampling week from 47 to 61. Overall, the expansion has added 504 samples, including zooplankton, to our sampling season. Let's dive into some average Longfin smelt CPUE maps for SLS. 2022 was a critical dry year. The goal is to capture the complete Longfin smelt distribution within the Delta. In a critical dry year, this is what that looks like. Notice the majority of the distribution in the middle near the confluence, and it somewhat tapers off as you go into Carquinez Strait. Now let's compare that to 2017, a wet year. A very wet year. Where are the Longfin? There's some at station 411 and there's some in the Napa, but the distribution is most likely outside of our sampling range for this year. 2019, another wet year. The highest CPUE is in the confluence area because we did not sample the Napa or San Pablo. Longfin smelt were most likely downstream and just outside of our sampling range again. Now this is 2023 without the expansion sites. It was a wet year and we can see high CPUEs in Suisun Bay leading into Carquinez Strait and also in the Napa. And when you add in the expansion stations, you get a better view of the distribution. Otherwise, we're only getting a fraction of the story. 2024, also above normal water year. It tells a similar story. The distribution of Longfin smelt are further downstream. And then I just like to toggle between them. This is what we're missing out when we don't sample the San Pablo region during wet years. Okay, a quick summary of SLS data. The figure to the left shows the total catch of Longfin smelt and SLS, broken up by routine, South and Central stations, Napa, and the San Pablo expansion sites. I just wanted to point out that the Napa has not always been a region we sample in. We started in 2014, sampled through 2018, stopped, and then restarted in 2022. But now I want to draw your attention to the last two bars on our graph for years 2023 and 2024. The green is the Longfin smelt catch at the expansion sites and accounts for 23% of the total Longfin smelt catch for 2023. For 2024, the expansion sites account for 16 of the total Longfin smelt catch. And fun fact, for 2023, we collected, ID'd, and processed 200,902 fish. 49% of that are solely from the expansion sites. Let me add some emphasis. We added 14 stations. 14 stations per sampling week accounts for 49% of total catch for 2023. Amazing. All right, let's dive into the some 20 millimeter Longfin smelt CPUE averages. 2017, a wet year similar to SLS, the distribution is westward with the highest average CPUEs at the high outflow stations and the Napa. 2019, another wet year, we see a similar pattern as 2017. The highest averages are westward. Again, this pattern for 2022 parallels SLS as the highest CPUE is centered near the lower Sacramento River. The distribution is more inland, typical of a drier year. 2023, without the expansion, it's a wet year, again, strongly resembles SLS. When you add in those expansion stations, you just get a clearer picture of the distribution. It tapers off as it goes towards 306. A quick overview of the 20 millimeter data. During high outflow years, we used to sample 5 stations in San Pablo. 4 out of the 5 are now part of our expansion stations. Let's take a quick look at 2023. The green shows the portion of Longfin smelt captured from the San Pablo expansion, which accounts for 21%. Again, some fun facts. We collected, ID'd, and enumerated 70,644 Pacific herring. The expansion accounts for 70%. Some highlights. Just a couple neat things. We captured larval rockfish, California halibut, and ronquil for the first time at these expansion sites. The picture to the left is my new favorite little guy. It is a California halibut, and I think it's the most rock and roll fish I have ever seen. It looks like it has a mohawk. I love it. 10 out of 10. No critiques. We also captured a larval Delta Smelt, 9 millimeter on May 10th, 2023 at station 327. We captured one back in 2019, also in San Pablo Bay at the high outflow station 335. So just think about that, but also think about this fish right here. Challenges with the expansion. Sampling in new territory brings new challenges, weather conditions like fog and rough waters, loads and loads of herring. All these things delay sampling and processing. Of the total time processing for fish for SLS in 2023, about 43% of that time was spent on the expansion stations. In part, this high percentage is related to the picture on the right. In the 2023 SLS sampling season, one of the expansion site samples had 11,278 Pacific herring. Would anyone like to guess how many Longfin smelt were in the sample? Just, yes, you're right, it's 2. You got it. 2024 has been a lot smoother, no crazy amounts of Pacific herring, and the weather has been really nice. It feels like I'm tempting fate to say those words out loud, but it's been really good this far. Closing. The main takeaway is during wet years, the increased sampling downstream is integral to integral to capturing a more complete picture of the distribution for longfin smelt. And this increased sampling has the added advantage of improving our design-based abundance estimates. It's a win-win. 2023 SLS and 20 millimeter data is published online at the FTP site. 2024 SLS data will be published soon. And I just want to remind everyone of our fish distribution maps and length frequency graphs. The QR codes here link to our CDFW survey pages, which have links that lead to both features as well as the FTP site. And yeah, so this is our fish distribution map, and this is our length frequency maps, graphs. And then just a couple quick acknowledgements. Takes a village to do monitoring. Yeah, and if I have time for questions or if anyone has any questions, that's it.
Excellent presentation. Loved it. It looked like when you were comparing your catch to previous years, you know, you had Napa sampling in 2017 and then again more recently.
Yes.
And in 2017, it looked like you hardly caught any in the Napa area or any at all. And then the past 2 wet years, you got a ton. Do you have any idea why?
No, and it's for 2017, we only caught 79 longfin, which is wild. 79 is a very, very low number, and I'm not sure why we only caught that many. Sorry, I couldn't answer your question, but thank you
Fair
for
enough.
asking. Yes.
Thanks. Great presentation. I think both you and Morgan should receive kudos for being poster children for displaying how the IEP adapts and updates its sampling programs to meet current management needs. Can you talk a little bit about what that took and about the challenges in doing that? We get criticized for not being too adaptable, and I think this is a highlight for how this is happening. So just tell us a little bit about that.
Yeah, so in like November 2022, I was pulled into, hey, we need to increase sampling in the San Pablo region for these for the regions listed here. We want to increase the abundance estimate. We need to increase 6 to 15 toes in this region. And so I was pulled into like helping pick out stations. And then from there, I'm very focused on the logistical side of field work. And so I'm wrapping up, and maybe we can talk after. Sorry. Oh, I'm sorry.
Yeah, no, we could talk forever about this stuff, and I would love to. So definitely, where's the mouse not going? Oh no. Okay. All right, so I'm last up today. So second half of the Vanessa section of our session here. So anyway, I'm Vanessa Tobias. I'm a statistician at the Lodi Fish and Wildlife Office, and I'm the lead for the Longfin Smelt Life Cycle Model Project. So today I'm just going to give you an overview of what we're doing, where we're at with the life cycle project, and really there's 2 parts to this talk. So the first is we're going to talk a little bit about how we're doing collaboration and how we're bringing in the community to help us set up the life cycle model to make sure that it's a useful product for a wide range of folks who want to use it for different things. And then the second part is going to be our approach to building the model itself. So I'll give you a little bit of an update on where we're at with that in the data collection part. So of course I have to acknowledge a lot of people. So we've got a modeling team, which is made up of folks who have experience doing life cycle models, like for the Delta Smelt life cycle model. We've got Longfin Smelt experts, whole bunch of people on the modeling team. We also have an advisory team that the modeling team goes to for advice and to run stuff by them. We ask them questions, they give us good feedback. So there's a whole lot of people involved in this. And I also want to thank our funders. DWR has a contract for us for the Fish and Wildlife Service component of this. Okay, so starting with the project timeline. The question I get asked the most already about the life cycle model is it done yet? And I would love for that to be the case. I would love to have something to give you guys right now. We just started. So we are working on a lot of different pieces. It's a complicated project. And I'll point out that our contract is up in 2027, so we have some time. But this is a cartoon of our project timeline. And it's broken into a bunch of different steps. So starting with the model plan. So we started actually writing down what we were planning on doing, running it by our advisory team, and writing up a document. So all this stuff is kind of planned out already, which is great because it helps us figure out like what's the roadmap for this thing, and we can all agree on where we're going. So that was the first step. We also need to build the data sets that we'll use for the model. We'll actually write the modeling code, which is the pink part. And then the big thing at the bottom is facilitating the collaboration and the open science part of it. So that's kind of where I'll start with today. But just so you know, there's a lot of overlap, especially between the building the data sets and the model part. And that's because it's an iterative process, and we'll be getting feedback as we go. So where we're at right now, again, we're building the data sets, we're starting the modeling process, we're starting to set up what the model looks like. And like I said, I'll talk about that in the second part, those two pieces. We're really deep into the data discussions right now, which is kind of fun. But before I get into that, I want to tell you a little bit about the open science and the collaboration part, because anybody who knows me knows that's my jam, so I can't leave that part out. Okay, so open science and collaboration. So we also wrote this down. So I wrote down all the discussions that we had about how we are going to do the collaboration bit. Like I said, there's a ton of people involved in this. So it really even comes down to like, how do we collaborate between the people that are part of the team? Because you all know how hard it is when you've got people from the state and the feds trying to figure out even what software do we have that we can all use at the same time and make this stuff work. So we had discussions about that and figured this stuff out. So I wrote it down in a plan. Things that are in the plan include who is a member of the teams and how do they overlap? How are the teams going to communicate with each other and what are they responsible for? That's super important so that everyone knows what they're doing when they're in a specific meeting. What's the focus? Really helps a lot to know where folks are coming from and what their job is. We also included the FAIR principles. So making sure that our project, the data and the products are going to be findable, accessible, interoperable, and reusable. I put a little plus sign on there too, because in addition to the FAIR principles, we also want to make sure that we're protecting and crediting folks for their intellectual contributions. So things like the data producers and the folks who are rearranging the data should be getting credit for doing those things. We also have to think about, since I come from Fish and Wildlife Service, we have to think about the federal open science standards and policies. So we're following those as well as the FAIR standards, because there's some additional things. Like I mentioned before, we also thought about the tools that we need to support our projects. How are we going to do this collaborative thing? Like, what platform are we going to use? What's accessible to everyone? And so some of the things that we've decided so far, and there's the little thing you probably can't read that's like, oh, we might change this as we go, as needs change. But so right now we're working on GitHub with a closed repository because that meets the federal standards. It will be accessible eventually. Okay, so that's the open science and collaboration part. Actually, I will back up for just a second and say that for the FAIR principles, one of the other things that we're super interested in is the interoperability part. So we're talking to folks with who are doing other work on Longfin and trying to figure out how can we incorporate what you're working on into the life cycle model, trying to understand what else is going on so that we're not doing this in a vacuum. And if folks have got like models that could talk to our model, we want to talk to you. So coming soon, we'll be in touch or get in touch with me. Okay, so a little bit about the methods that we're using for the model. So this whole thing, I'm really pushing this causal design idea. And here's some textbooks if you're a methods geek like me. So the idea with causal design is that you're putting the science first. So we're starting with the ideas about like what's going on in the system, and we're putting that ahead of the statistics. So the statistics will support the science. That's the big idea here. So you state your knowledge ahead of time. Using the first step is to make a conceptual model, and then you use it for things like data collection and to design the stats so that it's really all of the pieces are supporting the science that you're doing. Okay, so as Anke mentioned earlier, IEP loves a conceptual model. So here are 6 examples from recent technical reports from IEP. It took me about 5 minutes to find these because they are everywhere. And the reason that we love conceptual models is because it helps us communicate, right? We can put down our scientific understanding into a diagram. We can state our assumptions in the diagram, and we can have a discussion about that. So they're really great for us as a collaborative group for IEP to use conceptual models. And you can see that there's a lot of different ways you can make a conceptual model, right? So there's boxes up there, there's even like pictures and things like that. There's maps. These can all be conceptual models. For a causal design idea, we need a specific kind of conceptual model. It's called the directed acyclic graph, or a DAG for short. Here's an example of a super simple one for Longfin. So the idea with a DAG is that you have nodes and you have arrows. The nodes represent like data that you have in the system or something that you're interested in. Maybe you don't have data for it, but it's an important piece. So it's like a noun or like a thing. And then the arrows are the connections between those things. So how one thing influences another. And the really cool thing about them is that they are non-parametric to begin with. So you don't have to say what the arrows look like. You don't have to say what the relationship between, say, temperature and Longfin abundance is. You just have to say that there is a relationship, and that's how you start your conversation, which is very easy to do, right? Or relatively easy. We can say what's related to what. There are arrows because they're directed, so it wouldn't make sense to say that Longfin abundance influences temperature. That's kind of nonsense. And we know that, so we know which way the arrow goes. Simple, right? And then if we really want to write out an equation, there's one at the bottom. So like the abundance is a function of the previous abundance and temperature. But you'll notice I didn't say what that function looks like. It's just a function. So that's a good way to start. And then eventually we'll use that to inform the model, and we can start saying what those functions look like. So I think these are kind of fun because we get to draw pictures and then argue about them. So, okay, so the quickest introduction to Longfin life history ever. So you guys, like a lot of folks here, have talked about Delta Smelt for a long time. So forget about Delta Smelt for a second. Longfin don't behave like deltas really. Think steelhead. That's a really simple analog. It's not the best. I'm sure steelhead people are going to be like, no, that's not right. Close enough. So it's better than Delta Smelt. So the reason that I say that they're similar is Longfin live for more than 1 year. They go out to the ocean, they're anadromous. Some of them even maybe don't go to the ocean. Some of them stay. So like steelhead, there's a little bit of plasticity there. And yeah, so they're just a little bit closer to steelhead. So really quick, because I'm going to show you, this is a DAG, a directed acyclic graph for the life cycle of Longfin. And I want to give a shout out to Laura Mitchell for, she's the one that actually put all these DAGs together in the beginning. So this is just for the life cycle. So this is what you would expect. We've got the nodes are the different life stages. You'll see larvae in there a bunch because there's a lot of things going on with larvae. So the blue squares are nodes, and then the green circles are just labeling the arrows that are between them, which are super short because there's so much to cram on this. So you can see that we've got all the life stages in there. Then we split it out into whether they go to the ocean or not. And then the pre-spawning adults, they're on the 2 ends because this really connects. It's really a circle. But anyway, so that's the beginnings of the life cycle model. So we're going to use that to design our data collection. This is a synthesis project, so we're not collecting new data. We're using existing monitoring data. And we're breaking that out by time points. So one of the things we learned super early on in the process is that since Longfin live for more than 1 year, we really need to number the months because like there's more than 1 June and we were getting confused. So we'll see the months are always labeled with a number. And then we started lining that up with the existing monitoring data. All right, so we actually started working on abundance indices, or hopefully eventually there'll be estimates, but for now we're calling these indices using the existing data. So there's 6 different surveys. We've got different indices for all of them. These are just an example from base study of what those look like. So these are things that Leo Polanski's working on, and folks have probably seen that a little bit. So these are spatially resolved. They're using ratio estimators. And again, these have they're abundance indices with confidence intervals on them. So that's helpful for us. The next part would be designing the statistical procedure. So we're going to use a state space model most likely for this, and I won't go into all the reasons why. But we're also going to start putting in covariates. So I showed you the abundance part of the DAG. Well, now things are going to get complicated because we're going to start thinking about how does the environment play into the life cycle of longfin. So this is, okay, you can't actually see it. So this is our first draft of a influence diagram with environmental variables. And you'll see that longfin, the abundance part is in the middle there. And then we put all the environmental variables around it, and it got really complicated really fast. And I bet if you stare at this like closely, you'll be like, I don't like that part. I want to move that, and that's okay. So we actually already have moved this around. We have a new draft of this, but this is the only one that's got everything all together. And one of the things we realized again is that month matters. So what time of the year you're in. So we actually broke these out in the new version. So each transition gets its own influence diagram or DAG. And so this is just one of the examples. There are 10 of them now. I'm not going to show you all 10. But you can see that at the top, we've got that the life cycle part, the longfin data goes in the top. So those are the nodes that relate to larvae and juveniles. And then the habitat attributes have their own nodes at the bottom. And you can see that like it gets complicated quickly. And this is one of the more simple ones really. But for instance, like you might be interested in how, like down the line, once we fit this model, you might be interested in like how does hydrology affect longfin and the abundance, moving forward. And so you can actually trace that through the graph. And even though it might be a complicated path, you can trace it and we can say what that looks like. So that's a really useful thing about doing this with these diagrams is that we can trace how the environmental variables will affect longfin going forward. And like I said, we have to agree on what this looks like. We have to figure out what our model looks like and agree on it as a group so that we can have some consensus around it. Otherwise, if you don't believe the model that produces the model, like the conceptual model that produces the mathematical model, you don't believe the whole thing. So that's where we're going with this now is we're revising and doing this with some iterations with our advisory team. So I started talking about this a little bit already, but about the management question. So one of the things that we as modelers asked our advisory team for was like, what do you want this model to do? What's important to you? What's the end goal? And our advisory team actually gave us 90 ideas. So that was actually why we started like going down the path of doing the DAGs is because how do you prioritize 90 ideas and how do you say what's more important than what? So the idea is to try to incorporate as much as we can about the system into the environmental part of the model. But we're also going to have to be realistic about what's possible mathematically. So is this all going to end up in the final model? Probably not. But we are in the beautiful plains of this is all going to be easy and we have lots of time. So that's where we're at right now. But that's how you start, right? So start big, edit down. But we have a lot of stuff that we're interested in doing. And that's kind of where we're at with this. There's so much more under the hood of this. So if you want details, come talk to me. I will be happy to talk more about that. But that's the broad overview of the life cycle model and where we're at and where we're headed. But it's a super exciting place to be. And I'm really, really happy about this project. So that's what I have. Okay. We are out of time. It's time for the break. So come talk to me after. Come talk to the other folks who presented if you have questions, and I'll give it back to Rosie.
All right. Yes, we're now going to break. 20-minute break. Be back here at 10:25 for the next session, which is my favorite title, So Many Fishes. All right, folks, we're going to get started. Sorry for the delay. We had some technical difficulties, very unfortunate technical difficulties. And so the first talk of the session, we are going to have to miss for now because the computer ate Will's presentation and he was very good and got it to us early. And then, but we're going to soldier on and hopefully we can find some time for him to give a reprise at a later date. And so I'll give it over to Claudia, who is the session chair of the So Many Fishes session.
Welcome to the So Many Fishes section. I'm Claudia Macfarlane. I'm a database manager for the Enhanced Delta Smelt Monitoring Program at the Lodi Fish and Wildlife Office. Our first talk is going to be by Andrew Veery about the automated fish tracking and enumeration in dynamic environments. Let's get going. Oh, I'm sorry. I'm out here.
It's okay.
How do you do this? You just drag it over. That was easy.
That works.
You need presenter view.
Yeah, I'll do presenter view for sure. Good morning, everybody. Just find the mouse. I got it. Yeah, I got it. Find it. Save button. Is this not presenter view?
Yes, no, it's because we had to
First time.
the other one out first.
Oh, something was taking over.
Yes, and we can go.
Okay. Good morning, everybody. My name is Andrew Veery. I am the image analyst with Kramer Fish Science, and the title of my presentation is Automated Fish Tracking and Enumeration in Dynamic Environments. So we're going to count fish. Just a little overview of the project. This is a diagram of our sampling platform. What this is a pontoon boat rigged up with several sensors and different meters. It collects different environmental variables, GPS location, depth information, as we travel continuously through our sample sites. And at the same time, beneath the boat, we have a net forward the bow where we, sort of funnel fish through as we're driving through our sites. And the fish you see on the left will go through the net in the sample system, and then it'll go past a box that includes 2 cameras that are GoPros, and then past that, it's another camera that collects images of the zooplankton, and the fish discharge out the stern of the ship. So let me just give you a quick video of, it's the issue we're dealing with here. This video, you'll notice several species of fish, the direction they swim in, and some of the debris and plant material we expect to see in these videos. So you see several fish swimming different directions, gonna get the idea. We're gonna be faced with a challenge here with counting them automatically, right? So let's state our issue. All right, so our fish in our system, they exhibit stochastic behaviors in both the speed and the direction in which they're swimming in. And as we continue sample continuously to these sites, we are subject to variation in turbidity, plant material, and other abiotic materials moving past our cameras. And so can we actually develop a cutting edge solution that is capable of enumerating and classifying the fish while we also leverage our costs, processing speed, and the accuracy of our models? So why do we collect this data? Why do we collect videos? For monitoring behavior, for stock assessments, and a little more important to Kramer Fish Science, it's to provide crucial data to evaluate the impacts of our restoration projects. So we want to collect videos before, the restoration, control, and then we want to collect video after, right? And see how we did. And the video is automated, to dramatically reduce costs in the overall time in between the analysis. So in the time in which we collect the data and, our analysts get the reports out at the end. And it completely eliminates human bias and error in the video review process. So on the right, you see a chart. It's steep, but the bar chart on the left is the total hours for one of our projects, where our total human review hours for that entire year for that one project, which came up to be 740 hours. And with an optimized end game model, counter fish tracker doing this all by itself, we estimate over 100 times the reduced time for that, down to something like 7, right? Just running scripts, moving files around. So how are we going to do this? Well, step 1, we got to detect where the fish are in the frame. So for that, we're going to use a detection model. Detection model will also give us the class of the fish, or the species, and the size of it. And then we need to reassociate detections between each frame, right? Because all the detection model is doing is telling us how many frames have fish in them and how many fish are in that frame, but they don't know if
two frames, that all the fish are actually the same from the previous frame, right? So we count that way, we're going to overcount thousands of times potentially. So we use a tracking algorithm. What that does is uses these features about the image, and it compares it to the detection from the previous frames or 2 frames ago, and it uses this association logic to say whether a fish is the same from a previous frame. So there's 2 detection models. There's 2 main types of detector models. Is a single stage, which is the one we'll be using, that I'll be using this project, describing some more. How this works is it takes the input image. So that's just a screenshot from our video that I showed you before on the left. Simultaneously, this divides it into a grid and creates this probability mask on probability map you see in the bottom. So which cells are most likely to contain a fish that we trained our model on and which class it is. So you can see it's got some different colors for the different species of fish there. And even the plants getting picked up too, but that might be at a low confidence, right? So we'll filter that out. And then simultaneously it's projecting these bounding boxes around the grids. And the model is then to say which of those several boxes best fits that the object we're trying to detect, right? And so you see in the output to the right, we have the 3 fish in the frame are successfully identified and boxed up, right? We have sunfish, bass, and then the other one's a fish. It's unidentifiable. There's no features that really say which one it is. And our outputs there in a text file is the class, the XY of the centroid of that box, how high the box is and how wide it is, and the confidence value of how sure our model is it belongs to that class. The other detector is a 2-stage, right? So that last one I said works in 1 stage, it passes through that image, divides it up. This other detector is a 2-stage. This I can explain. Like I said, that Zooplane camera is behind our GoPros, right? So that an image you'll see in the top left the top left graph there. How this works is it shoots a light back at a lens, and then what you get back is the shadows. So you're able to detect these really small objects, which are bugs, and a lot of bubbles too. If you look at the top right image, that there in the middle is a copepod, and it's about, using the conversion rate, it's about 11 pixels high, which converts to about 380 micrometers. You need these 2-step detectors to get these really small objects. A drawback of the single stage is that it really misses these super small objects. So we got to use these 2 different detectors in our 2 different systems, really, in order to get this data. But you can see in that bar chart, the speeds, right, comparison. So the FPS is about 35 at the single stage, and for that multi-stage where it actually goes over the image and uses these features to say, here's an object, and then that object gets detected as its own image, right? So it's going to do that. It's going to scan the image, and then hundreds or maybe tens, hundreds will get classified, whereas a single stage, it does it all at once. So this is way faster, but you sacrifice those small images, right? And it's a little less accurate, but it works out very well for us, for our project. All right, so we have a single stage detector. Now let's get into some tracking algorithms. The 3 I'm going to go over is Euclidean distance and IOU. Those are just using the information that our detector provides. So the bounding boxes and the centroids, the particle filter, which is a little more advanced. We'll get into it, and the deep sort algorithm. So for this, you need to understand your system to the best you can, the different motions, how things work, identify patterns and variances, and then you need to factor in how powerful your computers are. So Euclidean distance, the algorithms to the right, you're taking geometry, right? It's just a hypotenuse, I guess, right? This is extremely easy to implement and extremely computationally inexpensive, right? You can do tens of thousands of these operations in a second on a good computer. But the cons, it's completely dependent on those detections almost every single frame, right? We're just using that bounding box to either say the centroid's so close, so we'll say, yep, that's the same fish, or the overlap in that box that we drew. How much they overlap, we can say it's confident it's the same fish. But like I said, this will not work with multiple objects nearby, ID switches. So the model is saying it's one class, one frame, and another class, next frame. It's just not going to work. And when it goes behind objects or other fish, right? It just fails in several areas there. The next thing I'm going to go over is the particle filter. It's also called the Monte Carlo localization method. So this uses a lot more random sampling to sort of predict the future state of your object, which is our fish here. So we're going to use one detection from a detector to initialize this. So it's the first step. We're going to initialize and propagate the particles. So we'll create 100, 200 particles, and then we're going to apply to each of those particle a sort of like this function that we expect. The function that we can apply the particles so that we'll project them into the next frame where we think our fish is going to end up. So we introduce a lot of randomness here because we're not sure where this fish is going to end up in the next frame, right? It's under this boat, it may be scared, not know where to go. So we use standard deviations, min maxes. We really want to try to predict the best. Obviously, the more particles you create, the better chance you have that fish landing on it, but the more, resources you need, your computer. And the next frame, frame 2, those are the same particles, but your fish landed on the particle successfully. We apply these similarity functions. So for images, I like to use color. We compare histograms in some sorts. So a histogram of an image is in the bins, you have pixel intensities. It shows you how frequent certain colors are in the image. And using a distance function, we can say how well those particles match that fish from the frame before. We assign, and based off that, we assign weights to every single particle. And the particles with the low weights, we kind of get rid of to save resources. And the heavier weights, we select, and then we resample these particles. And then in the next frame, you can expect the fish may land on those red dots, and you see how we're tracking through these frames now. That just depends on, you need just one detection in the beginning, and the rest of it does it itself. So this is highly robust to nonlinear movements and occlusions, so fish. And does not depend, like I just said, on consistent detections, but this is difficult to implement and very difficult to tune. And this is very computationally expensive. So I've tried implementing that, just decided to go with something else. Then we're at deep sort now. So this is gonna depend on 2 major components, motion and appearance. Let me explain the motion. So the sort part of the word, this is an algorithm that's been around for a while, used in a lot of other tracking applications. This utilizes a common motion filter to predict the bounding boxes in the future frames, right? So where it's lost, you see in the bottom left, those blue tracks are successful detections, and then the smiley face goes behind it. Those red boxes are where this motion filter predicts that box is behind that green face, right? So eventually, when that blue box is a detection, gets in a future frame, it uses that IOU overlap to say, oh, that's the same fish that we were looking at before. And it's re-ID'd, we don't double count that fish. And the appearance vector utilizes this feature extractor of the classification model and then calculates similarity between the detections. So how these neural networks work is there's tons of layers, and each of those layers has these nodes, which is just hundreds and thousands of questions maybe, that are just knowledge of why the features that you're seeing belong to a class, right? So you take a class studying why you ID things as salmon, you pick out, your features of the fins, maybe this or that. You have that knowledge, but you're just looking at the features. That's what's happening here. You just take that part of the model where it extracts like the edges of the patterns and don't ask any more questions. You're just taking the features and you're comparing it to the detection from like the previous frame. So now you have this hyperdimensional space here to describe these features, and you're just comparing using another distance formula to compare that to previous detection. So now we have sort of like the space that describes maybe the edges of this fish. You can see here, it collapses matrix into something smaller, more easy to use, while still retaining the features of that fish. So we have the motion part of it, and now we have the appearance, and you sort of cake those 2 together, and now we have this tracker model, essentially. So what we're getting into now is a little bit of results. This is the detection, the single stage detection, and most parts of this deep sort algorithm I just explained, working in conjunction in that first video I showed you. While these boxes are red, the fish is not yet counted. It's not yet successfully tracked. It needs, 4, 5, 6 of those in a row successfully to then call it a fish. And I'll change the color of the box to blue once it's successfully tracked. So you see here, the occlusions handled it. Sometimes it's not perfect. Some of these teeny tiny fish get missed. When there's a lot of text at the same time, sometimes works a little It misses some, but this is a pretty good job, as you can see, of classifying and tracking these fish. In this next video, I've slowed this down three times. This is like a 10-second clip. I mean, 3-second clip, but 3-second clip, but it's going to be 10 seconds here. And it's going to show you how well this works when there's just not a lot of pandemonium and chaos and plants. This works very successfully. And these fish are moving fast. Like I said, this is slowed down 3 times. And so in that clip specifically, it's over 99% successful there. So that's most of the results I wanted to share with you. We're still early in implementing this in the production phase, so I don't have a ton of performance metrics of how it compares to human review over time, but we can expect shortly to be rolling those processes out to get this built out and into production. So looking ahead, we just retune, retrain, repeat till we're happy with something, and even then, a new model may come out. So this is just a never-ending iterative process, really. We optimize these models. There's technologies now you can trace over your model, and all the weights you can apply, you can change to a lighter number format. It sacrifices a little bit of accuracy, but makes these models faster. I want to automate the lengths. So we have 2 GoPros that are in stereo, and using those two GoPros, you can get automated lengths, and we want to upgrade cameras and upgrade our computers. Some acknowledgements, the Kramer Fish Group. There's a lot of folks, here working on that signaling platform project. So big thanks to all of them. And a few groups we have funding us, and I'll take any questions from the audience at this point. If there's no time. Sorry.
No, they were in order. Next, we have Grace Aringer from the UC Davis Genomic Variation Laboratory, talking about lampreys in California. Mitochondrial phylogenetic analysis reveals previously unrecognized lamprey diversity.
Thank you. I'm excited to talk to you all here about lamprey. I will share the results of a project I've been working on with Matt Campbell, Pascal Gertler, and Mandy Finger. So we used genetic phylogenetic analysis and found that we didn't know all we can know about the diversity of lamprey in California. So why should we care about these lamprey in California? They actually contribute a lot of benefits to their environment. During this larval phase where they're burrowed into the sediment and filter feeding, they participate in a lot of nutrient cycling. They maintain stream bed conditions, improve water quality. And then as they mature into their juvenile and adult life stages, they're a valuable prey item, and anadromous species deliver marine-derived nutrients, much like other anadromous salmonids you're familiar with. Also, they're a subsistence food and cultural resource for a lot of indigenous communities across the West Coast, including in California, and a cultural resource. They're also an ancient lineage with a really unique research potential. This timeline here by CritFix shows just how old they are relative to dinosaurs even. They've been around for 450 million years, and therefore serve a really unique research purpose in evolutionary biology and biomedical science. So there are some barriers to lamprey conservation and management, especially in California. We have limited knowledge about diversity, abundance, and distribution. There's a lack of awareness in the broader community and in management entities. We have inadequate historical data. The data we do have is geographically and temporally fragmented and not species specific. Additionally, there's taxonomic uncertainty. So there are a lot of questions about where species boundaries actually are within this lineage, and there's life history variation. Some are anadromous and some are freshwater resident, and we don't know how that life history variation should be classified as separate species or variants of one species. Because all of this taxonomy that exists today has been primarily based on morphology, genetics are a great tool to help us dig a little deeper and understand the diversity. So with that said, the overarching project goal was to characterize lamprey diversity. And this is important because it helps set management goals. For example, if there are migratory lamprey present, we have to allocate some resources towards fish passage, amongst other things. Whereas if we only have non-migratory lamprey, we might be able to allocate more resources to larval habitat. Additionally, as we all know, legislative frameworks require distinct population units in order to move forward with conservation action. And to characterize this diversity is fundamental for all future research. So the first part of this was to identify currently recognized species present at each sampled location. And to do this, we use genetic species ID. And to do that, we use a method called DNA barcoding. We basically take DNA from individual lamprey and sequence it and compare it to a public database to come up with a species ID. Second, we wanted to compare the current taxonomy, these species classifications based on morphology. How does that compare to the genetic patterns that we see? So to do that, we estimated molecular phylogenetic trees and performed what's known as molecular species delimitation analysis that just takes all the genetic data and parses it into species delimitation based on the genetic patterns. Last but not least, we wanted to see how this diversity is distributed across the landscape. Do we see it kind of randomly found throughout the state, or are there pockets of diversity in specific regions? So first, I wanted to just give a little bit of overview of the study. These are our sample locations. I do want to highlight that this study would not have been possible without a collaborative network of fish monitoring programs throughout the state. This project was made possible by opportunistic sampling, and we were able to collect samples from 19 sites across 15 subbasins. Most of the lamprey we collected were larval individuals, and at this stage, all species look virtually identical. So it's not surprising that over 50% of the samples were unidentified even to the genus level. So we took a fin clip, extracted DNA, and sequenced what's called the cytochrome B gene. You don't need to know anything about the gene. It is widely used for phylogenetics. And there are a lot of lamprey sequences for this gene in these publicly available databases, which is key for the barcoding approach we used. To give you an idea of what we expected to find, these are the currently recognized species that we expected in the study area. I do want to point out that these two species grayed out here, we don't know very much about. For each of these, there's like one paper describing these species, and then they've never really been found or recorded again. And there aren't sequences in the database. So we're not going to talk a lot about those two species, but I do want you to remember that there are 5 Entrainment species described and 3 Lampetra species described. As you can also see here, there is diversity in migratory behavior and feeding behavior. Some are parasitic, some are not. They are all species of special concern in California. Peter Moyle in 2015 indicated that these are all species of moderate or high concern. To give you a little bit of a better visual of what the species diversity looks like, here are the 3 migratory lamprey we find in California. They are different in body size, and they have different oral disc dentition patterns that are the most diagnostic features to distinguish between them. But again, these features are only present in the adult stage. The rest of these species are freshwater resident brook lamprey. They are non-parasitic, and their oral discs are a little bit underdeveloped. The teeth are less sharp. But again, these distinguishing features are only present in the adult phase. So to get into our first goal, which species are present at each sampled site, we use this method called DNA barcoding. And this utilizes mismatches in DNA sequences between individuals And if you sequence a number of individuals at a gene that's long enough, you get a pattern that looks like this, and it starts to look like a barcode. So then you can scan that barcode, so to speak, compare it to the database, and identify what species is present. So these are our results for Lampetra. In yellow, this is the expected range of Western River lamprey, Lampetra ayresii, and the black dots represent sites where we did indeed find that species. Similarly, in pink salmon color, that is the distribution of Kern Brook lamprey. And that single green dot in the Kings River is the site where we found the Kern Brook lamprey. Similarly, in Entus venus, Pacific lamprey, the species you all probably are more familiar with, is the most widely distributed and most abundant sample that we found. All of the black dots, including these black outlined purple dots in the Klamath, are sites where we found Pacific lamprey. Klamath Brook lamprey were found at 3 sites in the Pitt River and Goose Lake Basin, Lassen Creek. And then last but not least, the Klamath River lamprey was found, as expected, only in the Klamath. But I do want to point out that there is a significant range overlap between the Klamath River and Pacific lamprey. Just highlighting that species ID is really important when we're encountering these lamprey in the field. So to summarize, we found Pacific lamprey at 14 sites, Western River lamprey at 7 sites, mostly in the Sacramento Basin. And then these other 3 are known to have more geographically restricted ranges, so it's not surprising we found them at 3, 2, and 1 site. Moving on to our second aim, we wanted to compare the current taxonomy to the patterns that we found. So again, in Lampetra, there are really 2 recognized species, 3 maximum, but there's debate. And in Entus venus, 5 recognized species. Now, this tree is really hard to look at. It's tiny, and I don't want you to really try to read it. But what I do want to highlight is that within Lampetra, we see these really deep divergent branches. So there are distinct clades and a lot of genetic differentiation between these groups, while in Entus venus, you can see you don't really see any branching. Everything's really shallow. Everything's really closely related to each other. And I want to highlight that at the genus level, the taxonomy is accurate. So everything that we thought was Lampetra is indeed Lampetra, and vice versa. This pattern is also highlighted in our genetic distance measures, which is shown in this heat map table here. Different lineages within Lampetra have this moderate salmon colored genetic distance, indicating that these lineages within this genus are really different from one another. While in Entus venus, these really low levels of genetic differentiation show that even these distinct lineages are really similar to one another. So looking a little more closely at Lampetra alone, again, this tree is kind of challenging to read. I will make it simpler in just a minute. But this is where we did the molecular species delimitation, and these are our outputs. Here, a unique color represents a distinct species that was indicated by this analysis. We use multiple methods, and the most highly supported outputs are shown. Another thing I want to highlight is that we pulled in publicly available sequence data from a study done in 2012 by Boguski et al. To help cover some more geographic ranges that we weren't able to sample. So to simplify this a little bit, I've just taken that consensus color from the species delimitation results and indicated it on the tree here so it's easier to read. So we have our 2 recognized species, the Western River lamprey, and it seems that there are 2 distinct populations on the North Coast and in the Central Valley. And then what gets really interesting is when we look outside of these currently recognized species, we found that there are new species that have not been described in Alameda Creek, in the Russian River, and the Napa River, in Clear Lake and possibly in Paynes Creek, although that was based on a single sample. So we need a lot more research in that area to validate those results. So the Russian River, Paynes Creek, and Clear Lake samples were from a previous study, but these populations in the Alameda Creek and Napa River had never been sampled or indicated that there was morphological differentiation. So it was really exciting to find that there are these two new species in the broader Bay Area that were previously unknown. In Entrainment venus, things were a little bit more like what we expected. There are 3 currently recognized species indicated in gray, yellow, and orange here. We attempted molecular species delimitation, but all the programs performed very poorly. The analysis basically failed. And they were unable to delimit the currently recognized species, likely due to the high sequence similarity. So to put this on a map, we see throughout the Central Valley and on the North Coast, we see Pacific lamprey, all genetically very, very similar. And then we do see some distinct populations that are known as species, the Pitt Klamath Brook lamprey and the Klamath River lamprey. But there's really low genetic differentiation, in fact, lower than what we would expect between species. We're a little short on time, so I'm going to breeze through this last bit. How the diversity is distributed across the landscape is actually similar to these patterns of native fish diversity that have been observed in other lineages. These are zoogeographic provinces defined by Peter Moyle in 2002. And if we overlay our results on top of this, we can see that in Lampetra, there's actually more diversity in the Central Valley than expected based on these patterns observed in other fish taxa. It is the largest, so maybe it's not too shocking. But other than that, there are lineages like in the North Coast that we expect, and in Clear Lake that are distinct and follow other fish taxa patterns. In Entrainment venus, we see even more, the lamprey follow even more closely the pattern we see in other native fish taxa. In the Pitt River and Goose Lake Basin, there are distinct populations, as well as in the Klamath, not in the North Coast. But again, that's not too shocking. So to conclude, there's really high lamprey diversity in California. I think most people know about the Pacific lamprey, but don't really know about these other species. Multiple species co-occur at many sites, and we see really high genetic differentiation in Lampetra, low genetic differentiation in Entrainment venus, and the current taxonomy is not sufficient to describe all of this diversity. And the geographic distribution is similar to other native fish taxa. My time is up, so thank you, everyone. Feel free to take a picture and ask me questions via email since we don't have time here. And thank you all for coming and listening.
Thank you, Grace. We all love our lamprey friends. Next, we have Levi Lewis from UC Davis talking about otolith-based insights and management implications for Delta Smelt.
Thank you. Finally, a smelt talk. Back to smelt. My name is Levi Lewis. I'm the director of the Otolith Geochemistry and Fish Ecology Laboratory at UC Davis. We use otolith-based tools to look at the life history traits, demographic traits of endangered fishes, and we also do surveys of fishes in wetland habitats. Today, what I'd like to do is provide just a high-level overview of a bunch of work that we've been doing using otoliths to better understand Delta Smelt demographic traits. And what I really want to try to achieve is to talk about what we found, what does it mean, and why it might matter. First, I want to acknowledge a lot of my co-authors that are up there. There are many other individuals that have contributed to this work, and I just can't list them all. Funding came from the U.S. Bureau of Reclamation, the Directed Outflow Program or project, and the specimens came from U.S. Fish and Wildlife Service, EDSM, and CDFW. And I'm sure I'm missing people, but those are the main folks involved in these studies. So you know a lot about Delta Smelt already. They have a 1-year life cycle. They've been described as anadromous migrants within this estuarine system in the upper part of the SFE. However, distribution data suggests that maybe that's not quite right, and we'll touch on that more in a moment. This small anadromous fish was one of the most important components of pelagic food webs, and it is no more. So now it's critically endangered due to many different possible factors. I won't list them all, but they're shown there. One of the ones of main concern that we deal with in this group are the effects of water exports. And it's been really important to try and think about how exports are interacting with all the other stressors to cause the decline of this fish and what we can do to try and stabilize and reverse it. Also, this has created conflict both locally and even nationally, with Delta Smelt showing up on Fox News and the halls of Congress and even spewing from the lips of past presidents. And so the agencies and managers have really been trying to get a better grasp on the ecology and biology of this fish in order to be able to provide new tools that we can use to provide water to agriculture, industry, and municipalities, while also trying to protect our natural heritage. Delta Smelt live nowhere else. They are California. So there's been a lot of funding going into various different ways to do this, looking at distributional data and environmental data, doing lab experiments with cultured fish. And what we've been funded to do over the first Delta Smelt paper came out in 2007, but recently with the DOP over the last 5 years is to use otolith-based tools to really understand what's going on in the wild population. So before we do that, we had to develop methods, and I'll just briefly show what that looks like. Sorry, I think I double clicked there. Anyways, so we've used a variety of different validation studies, partnering with a fish conservation and cultural laboratory. This is a result of one of those validation studies where we received a bunch of known age Delta Smelt, and we looked at how well could multiple different agers, many of them undergrads, but trained, how well they could reconstruct the known age of the fish. And you can see that line is pretty tight, and our accuracy and precision was pretty good. So we're pretty comfortable that the otoliths are providing really good age data on Delta Smelt. What about growth rates? Well, we can also use the increments in otoliths, and those increments the increment widths can tell us something about growth, but we want to confirm that that's actually true. We did an experiment. We can see the pictures above where we had fed fish and unfed fish, just thus forcing differences in growth rate. And we looked at when we were able to detect that using otolith-based techniques. And so in this figure, you see the difference between fed and unfed treatments. And for the first 150 days, you don't see really any major differences. And out of 153 days, when we imposed those feeding restrictions, we see a dramatic change in the growth rates of the otoliths in those fish, which corresponds with the changes in the growth rates of the actual fish themselves. And also using otoliths to track migration. We used otolith geochemistry. I won't go into the details. Here I'm showing strontium isotopes. And in the left plot, that's how strontium isotopes, which are on the x-axis, vary in relation to salinity in the water. And we can use that to generate the model on the right, which allows us to examine how the salinity, the known salinity on the x-axis corresponds to the estimated salinity on the y-axis. And you can see that the error or the confidence, the error goes up and the confidence goes down in salinity estimates as we get above 6 to 10 ppt. And so we're really focusing on low salinity habitats, which is perfect because that's where Delta Smelt live. So we did an experiment where we had fish in the FCCL and we manipulated salinity. So we have a treatment where fish are at 0 ppt and another group that moved to 3 ppt and 6 ppt. And then we looked at how well we can reconstruct those salinities of those fish with their known histories. And as you can see, it's pretty good. And not only that, but the timing of that first transition from 0 to 3, again, in the low salinity transition, we got to it within 6 days. So I hope that those methods development studies, that's a body of work on its own, but I hope that it convinces you, as it has convinced me, because I'm very skeptical, that these tools are actually pretty good for looking at Delta Smelt traits. But that's not really what I wanted to talk to you about. What I really wanted to focus on is the application of these tools to better understand the wild population. So with respect to growth, we found that growth in wild Delta Smelt is quite sensitive to environmental variation. And this confirms expectations from a bunch of different laboratory-based studies with cultured fish and distribution studies based on all the surveys that folks have been talking about in these sessions. So with respect to temperature, we found that growth declines, maybe even at lower temperatures than we would expect at 18 degrees Celsius. We found that fish that are in brackish conditions are growing better than fish that are in freshwater conditions. And again, that supports several other studies, including others of ours. And fish are growing better when it's turbid, so when clarity is low. None of this is actually surprising, but what's really interesting is that this is the first time we demonstrated these hypothesized results in the wild population. We've also examined phenology, or the timing of events in a population. And the way we did that is that we used the hatch date. We used the capture date and we subtracted the age of the fish to get the hatch date. And then we looked at the patterns in the median hatch dates of multiple cohorts and how that relates to environmental conditions, specifically winter temperature. So on the x-axis, I have winter temperature, and on the y-axis, I have the median hatch date of each of multiple cohorts of Delta Smelt across different years. And the hatch dates span from roughly March at the earliest to maybe early June. And what we found is that hatch date is strongly, significantly correlated with winter temperatures. And so phenology, like growth, is sensitive to variation in environmental conditions, sensitive to variation in climate. We've also been using otolith geochemistry to retrace the life history of these fish. This first plot shows just the distance across an otolith. So that's the life history of a fish from birth until death. And the y-axis is showing the strontium isotope ratio. And you can see there's quite a spread of different patterns, indicating that this fish is doing a lot of different things. We can use some classification tools to separate those out into at least 3 different life histories, which are migrants or MIG, freshwater residents, FWR, brackish water residents, BWR. And so Delta Smelt has a portfolio of behaviors that it uses. It is not just a migratory fish that moves from freshwater to saltwater like a salmon or like a steelhead or, as Vanessa pointed out, or a steelhead. Delta Smelt are really staying up in that estuary and doing lots of different things up in the upper estuary. And so this is what we're kind of proposing of the Delta Smelt life history, and I'll mention why that matters and how it relates to our understanding of life history nomenclature shortly. But not only that, what we've also found is that the relative proportion of those different life histories varies significantly across years. And not only does it differ or vary interannually, but it varies interannually in relation to the environmental conditions or the climate of that year. This is the result of a multinomial logistic regression, which was looking at the relative proportions of the different life histories across years as a function of 2 main environmental drivers, 1, outflow, and 2, delta temperature. And on the left plot, it shows the proportion of freshwater residents, and where that high peak is, that's about 50% of the population being freshwater residents. That occurs when flow is low and temperatures are really cool. And we see the opposite pattern with migrants. When you have really high outflow and really warm water, you get more migrants. I can explain in depth why we think we're finding that. I don't have time, but I would love to talk more about it. Not only are we finding that climate is driving life history, but we're also finding that those life history patterns, when we combine it with genomic data from the Genomic Variation Lab, specifically Mandy Finger and Matthew Campbell, we find that the life history patterns are genetically linked, and we can use the genome of the fish to classify an individual into a migrant or a freshwater resident with 95% confidence. So these are not just random life histories, but there seems to be some sort of potential heritability tied to this. And last, not only are we seeing that, but we're also finding that the life history determines the fecundity of these fish and the size of these fish and the growth rates of these fish. So for example, this is work by my postdoc, Leticia Covole, who isn't here because she's moving back to Brazil now, but I'm sure she's here in spirit and hopefully she's listening. She's done a wonderful job working with Bruce Hammock's lab and Sweet Tay's lab to examine how life history relates to the fitness of a fish. And what these data are showing is that fishes that are rearing in saltier or brackish conditions for most of their lives or migrating down to those habitats have higher body mass, they have higher gonad mass, and they have much, much higher fecundity. So life history is also directly tied to fitness. Okay, as I mentioned, those are all very high level kind of overviews of that body of work. Basically, what I want to focus on is that these data are really emphasizing the sensitivity of this fish to climate. And these data are really emphasizing the potential genetic component to that life history variation and how it influences the fitness of Delta Smelt. And to my knowledge, and I'm happy to be corrected, I don't think any of this information has really been incorporated into any of our models or our efforts to manage the species. So now I'm going to get into the dangerous part of this talk, which is to start musing about what I think personally. And these are just thoughts, don't hold me accountable. I know it's being recorded. These are really my musings and some ideas to hopefully, spurn some additional conversations about what we can do to protect this fish. So first, The first theme is about Delta Smelt life history and habitats. Like, what does it need? And the first is just the nomenclature. I would argue that Delta Smelt should not be called anadromous or even semi-anadromous. They're really different from a Longfin smelt, as Vanessa pointed out. So we shouldn't be grouping them together. They do something different, and they also have complex life histories, and we should really say it and acknowledge it when we're talking about this fish. So in the literature, they might be called a freshwater estuarine opportunist, really emphasizing that freshwater linkage, which is why they're so susceptible to what we do in the Delta, right? And if you look at our new conceptual model for Delta Smelt in terms of their life history, you can see those two look quite similar. Two, we really need to think about the likely importance of freshwater habitats. I know I don't need to say that to this group, but really emphasizing that just the migration itself is not the only thing keeping this population around. The freshwater resident component of this population over evolutionary time likely was really critical for its long-term survival. We know that that's true for other fishes like lamprey, and we know that's likely true for other fishes like steelhead. And that's probably an important component of the life history of Delta Smelt. And therefore, the loss of habitats in the Delta, which in large part have been kind of ignored, and their year-round impacts might be underappreciated because we assume they're leaving and they're migratory. Okay, and then the third part of this theme is that we might have some issues of shifting baselines, or what they call ecological amnesia, with respect to what this fish does and what it needs. So everything that we know about Delta Smelt is based on the last 60 years, maybe even more, the last 20 years of data collection. But this fish has been around for a long time, and the conditions at which we're studying this fish are really different than the conditions under which it evolved. And so the ecological and life history strategies that it used may be maladapted now to what's happening, and our understanding of what it needs may also be a little bit misplaced. So for example, what we know has been during this period of incredible warming, and but prior to that, Delta Smelt had 4,000, many more thousands of generations in a completely different system. Again, where it was cooler, and there might have been a huge portion of freshwater residency that was adding stability to that population. Okay, and then the next theme is that climate change is a major problem. I do not need to belabor this point, but the key thing I want to note is that I don't think there's a future for Delta Smelt without addressing it directly. Growth, fecundity, life history, and fitness are all affected by this, and that we probably need big, bold options and actions to deal with it, including temperature management, assisted migration, which has been described and which has saved sacramento perch and the Lahontan cutthroat trout, and has been described by TJ O'Rear many times, and even assisted evolution, as I believe Joanna even talked about. And then the third is that life history diversity is really important. We know that's true for many other species. And in the hatchery, we're currently producing 100% freshwater residents and then putting them out in the environment, whereas 80 to 90% of the wild population was migratory. That's the life history that it actually used. And that mismatch between conservation and ecology is something that we should probably talk about. We also have mismatches in phenology that we should discuss. And we should be concerned because now there's directional selection every year away from the dominant, the fittest, and the most fecund life history. And I don't know if that's really being discussed. And I just mentioned how awesome migrants are. So I'm gonna stop here. I know my time is short. And these are kind of those conclusions just summarized yet again. And I'm happy to leave it up if we have time for questions. We have one here and then, yeah, happy to.
Thanks for that talk, Levi.
Sure.
I was just confused a little bit by that last point. If the migratory strategy is somewhat heritable, how can we say that 100% of the hatchery fish are freshwater residents?
That's a really good question. And we have to talk about phenotypes and genotypes, right? 100% of the hatchery fish that are being released have a freshwater resident phenotype because that's how we rear them in the hatchery. And over time, you can select out migratory genes if you push that and you have an annual fish every single year. That was, yeah, thank you for that clarification. Do we have time for one more?
You can stand up. I just had I was a little uncertain
Sure.
about your what your winter temperature is and also your flow. So is it water temperature, air temperature? What is the period of the winter? And what were the units on your outflow?
Yeah, that's a really good question. So we used for that study, we've done a couple different analyses. For that study, if I recall correctly, we used the winter temperature index, which is the mean delta temperature based on a series of selected sondes over the winter period, such as like December, January, February, March, if I recall correctly. And similarly with flow, we were using a summertime migratory flow, which was later on in the life history. I can send you the paper if you'd like to see it, but I know we're short on time. Thank you.
Thank you. Next, we have Jonathan Walter from UC Davis talking about environmental drivers of fish populations in Central California.
Beautiful. Thanks very much. My name is John Walter. I'm a relatively newly minted PI in the Watershed Center at UC Davis. And I am a statistical ecologist. I use data science, mathematical modeling, statistics to better understand the dynamics of aquatic biodiversity and ecosystems. So to briefly contextualize the work I'm going to share with you today, there's deep concern for fish populations in California. I think the many talks at this workshop helped to bring that to the fore. But in this context, there's been a lot of attention paid to a handful of species that are really already known to be in pretty dire straits. But we also have, in part driven by those conservation concerns, have these long-running investments in monitoring that provide valuable data on many other species whose dynamics and environmental associations are not so well documented. Although I do want to acknowledge that there's often internal knowledge of some of these species and data sets that isn't always well reflected in the published literature. And as someone kind of new to this system, I'm learning about that and will continue to learn more, I think, as I get deeper into this role. So the objectives for this study was that for a suite of 21 relatively common and abundant fishes, we wanted to characterize some long-term interannual patterns in recruitment. We wanted to evaluate how those patterns are related to a range of environmental variables that are representing putative drivers of populations in the system. And we also wanted to quantify how different data sources relate to the modeled patterns as a means of better understanding how different monitoring programs that have different designs and objectives actually contribute to providing complementary or contrasting information about these populations. So for data, we used a selection from this product that was developed by Beshevkin and colleagues, a nice example of how the IEP has produced publicly available data that can support a lot of interesting work. And it was a huge lift to do all of this work, I'm sure. So thanks for that. So we focused in this study on some of the 4 longest running programs and used that to produce 1980 to 2020 time series of annual fall age 0 abundance. We use this as an index of recruitment. And for each species, we analyzed aggregated time series for unique region, monitoring program, and sampling method combinations, where those sort of combinations provided adequate data to be used in model fitting. So along the way, we developed a set of maximum length thresholds for age 0 fish by species and monitoring program and month. Although there were some existing thresholds and knowledge of this available, the ones that we could access didn't cover the complete set of species that we wanted to consider in this work. So we went ahead and developed our own based on a combination of information, including some existing thresholds that were used by the base study, interpreting histograms like you see behind me, and then a quantitative technique called normal mixture modeling. And then just to provide an example, for this starry flounder using base study data in the month of November, we could kind of set our threshold at this trough in between peaks in this length frequency histogram. This has a nice clear bimodality. Of course, not all of them were quite so clean, but just to give you an idea of how this sort of thing works. And then here are time series for the set of environmental covariates that we considered. Delta inflow was included to represent sort of an aggregate of some upstream conditions and might be especially important for some more freshwater associated species in this pool. Several variables that were supported by various programs for environmental monitoring represent different delta and estuary conditions. And then we also included some a pair of coastal ocean variables. And these were included because some of these species are also using marine habitats. And as we know from some prior work by Jim Clarn and others, there is some pretty strong correlations, especially near the mouth of the bay, between ocean conditions and the estuary conditions. So we analyzed these data using multivariate autoregressive state space models. I'm gonna totally gloss over the maths here, but just to kind of unpack some of the key features of this approach. Multivariate just reflects that our response data are multidimensional, these being time series from different combinations of region, monitoring program, and sampling method. And we considered these to be independent records of overall population trends. Autoregressive just means that the values of our response variable, this age zero abundance index, depend on prior values. And then state space means that we had explicit models of both fish population change and the observation process. And really explicitly modeling the observation process helps us to deal with variation that's associated in different practices between the monitoring programs that isn't really explaining the dynamics of these populations. And then we use information theoretic model selection methods to tell us which environmental variable or no environmental variable from that candidate set that you saw previously best explain the dynamics of each species. So finally, when I get to some results here, so here are some modeled trends in age zero abundance for the 21 species that we successfully modeled. And these represent a single trend shared across regions where there was sufficient data to model them. And then these are also organized roughly so that more marine associated species are toward the top of this graph, and then more freshwater associated species are toward the bottom. And then significant increasing trends are in blue and significant decreasing ones are in red. So one thing that probably stands out to folks is the preponderance of species that inhabit more marine to brackish salinities. And one other thing that's sort of comfort here is that our approach to handling these data for some of these better known, better studied species like striped bass, like longfin smelt, like Delta smelt, show long-term trends that accord well with what we expect. So we probably haven't done something really stupid with these data. This figure summarizes how each of these species are related to environmental drivers. We considered models with no or one environmental driver. The color in this heat map indicates the AIC weight, and this can be interpreted as the probability that a model is best in its group. And the top model for each species is identified with a star if the no covariate model outperformed all the others. I'm sorry, this figure is looking a bit darker on the screen. It's a little bit more difficult to read. But there are plus or minus signs indicating the direction of the relationship with the top covariate. A few highlights that I wanted to raise from this. So water temperatures in the delta were the most commonly selected top covariate, and across all of those species, they were negatively associated with warmer water temperatures. If we look at that time series for delta water temperatures, we don't really see strong long-term trends in those data. But if you do think about the impacts of climate change, we can expect warmer atmospheric conditions, possibly also periods of lower flows, and all else being equal, we would expect those things to translate to warmer water temperatures. So I think this result really reinforces concern for what the fish assemblage may look like in the future. One other point, plenty of previous work has documented increases in water clarity and lower pelagic primary production in the system. And this analysis suggests mixed effects on different species. And then lastly, we did see some statistical relationships where there is not a plausible direct mechanistic effect. And notably, this is this trio of more freshwater associated species that were statistically related to ocean variables. This is likely explained through relationships between ocean conditions and large-scale climate that also impact inland aquatic habitats. And it's also a good reminder that this is a correlative study with those kind of inherent limitations to it. The last piece of results I want to share is this figure that represents the correlation between the modeled age zero abundances and the unique input time series from different combinations of data source, sampling year, and region. And white indicates a combination that wasn't used due to insufficient data. The blues indicate positive correlations, and the red indicates negative correlations. And the big takeaway here is the preponderance of blue in this figure. And this indicates consistency among data sources in the representation of interannual variation in recruitment. To kind of begin wrapping these results up, we see a diversity of positive and negative trends, and this is kind of consistent with a reorganization of the fish assemblage. And these were also shared among native and non-native species about in proportion to their prevalence in this data set. That diversity of trends was also underpinned by a diversity of relationships with environmental drivers. And we didn't really see evidence that species that shared a driver exhibited similar long-term trends. But we did see many species negatively associated with delta water temperatures. And this is likely a key variable to think about considering climate change. And then lastly, we saw that different monitoring programs and gears were generally providing consistent information about fluctuations and trends. It's not necessarily the case for true abundances. We know that different gears have different biases, but for interannual variability, they tend to track each other in consistent ways, at least for this set of species. There were a number of species, a handful of species that sort of met our criteria for commonness and abundance, but weren't successfully modeled. And in general, these were species where there was greater disagreement between different regions, different gear types in what those populations were doing. They didn't fit very well the assumptions of our modeling framework, and so we left them out. But that's one caveat. So our next steps for this work are to quantify potential indicators of instability of these populations. It'd be a real benefit to know what species are at risk before their populations exhibit signs of crashing. And traditional methods for doing that, like population viability analyses and some of the modeling efforts that you saw presented on earlier, require data that aren't readily available for many of these lesser studied species. And the gears that we use in these programs are generally best for smaller juvenile fishes, leaving out information about the larger and older fish. So we're going to apply a selection of some generic statistical indicators of stability and regime shifts as an alternative approach to investigating risk of collapse given these available data. And one example of this is seen at the bottom of this slide is for Longfin smelt. On the bottom is a population trend, I believe from fall midwater trawl data. And on the top in blue is a time series of the lag1 autocorrelation coefficient. And this is one of these generic stability indicators with increases potentially indicating loss of stability. We see this ticking up as we see populations of longfin tracking down. So that's all for now. Thank you very much for listening, especially for CDFW for funding. So folks that can provided feedback on earlier versions of this work. And I think I might have like a hot second. Cool. Thank you very much.
I'm sorry if I missed this. Were you only looking at the fall months?
Yes.
Okay. Are you at all concerned that there's a lot of seasonal variability? Like a ton of seasonal variability. And so I think if you wanted to expand on this, you should look at trends throughout the year, especially there are many species that are young in the spring as well.
Yeah, thanks for that. Yeah. Certainly that's the case. There's a good deal of variability. We wanted to focus on the fall months and restrict this to age 0 to try and sort of control for that, to have sort of one common thing that we could apply across species. Of course, it's the case that it's not going to be ideal for every species in this set, but it was an attempt that we made to try and get something that could be comparable across a broad set of species. But yeah, thanks for that. Cool. Thanks so much.
Thank you, Jonathan. Lastly, we have Scott Meyer talking about biofouling impacts to field enclosure studies on Delta Smelt.
All right, thanks for tuning in. I'll promise to keep it short so we can all get to lunch. But thanks for tuning in. I'm Scott Meyer. I'm with the GWR. Today I'll be presenting the work that we did on the effects of biofouling on field enclosures on Delta Smelt. So I'll start off with a quick introduction of the field enclosures themselves. So these enclosures were designed by the Fish Physiology Lab at UC Davis to conduct in situ experiments with Delta Smelt. So this process was pretty in-depth. Everything from the mesh size to the angle of the floats was considered when making these. And so we got the enclosures and we started using them. And over time, we started to observe something that kind of concerned us, and that might be like confounding the effects of the work that we do, and that was biofouling. So what we're calling biofouling is just this buildup of organic material on the enclosures, mostly made up of algae and macroinvertebrates. So a big concern would be like that the algae is growing on those enclosures and causing water, like keeping water from exchanging in and out of the enclosures. So that could affect the water quality and the ability of the smelt to be in there. Also, all of those macroinvertebrates that grow in the algae, we were concerned that the smelt would be picking those off and eating those, and that's subsidizing their diet and keeping them healthy, and something of that otherwise shouldn't be the case. So we set out to investigate that biofouling, and with these kind of research questions in mind. So first off, we wanted to see if we were even able to eliminate or reduce the biofouling in the field. So this is a problem in the summer and the fall. In the winter, this biofouling really doesn't grow, but in the summer, fall, it can grow pretty quickly. So, we can't go out there every day and clean up those cage enclosures. And so we wanted to make sure that we were able to do this feasibly. And then the second question was that, do the smelt held in the enclosures exhibit a difference between two different field sites? So like a big question that we want to use with these enclosures for is to answer like if the smelt are able to response from the Delta smelt held in the enclosures, is that so like if we see a difference in the condition of the smelt, is that because of the biofouling or because of the habitat and location that they're being held at? And so we wanted to use two different field sites to test that out. And so our first method here is to come up with two techniques to test out. So our first was to just scrub the enclosures every week. So it's pretty simple. We never thought to take any pictures, so I don't have any fun graphics. But we just went out every week and we scrubbed the outside of each enclosure. We didn't scrub the inside because we didn't want to like cause undue harm and stress to the smelt. And then the second was to replace the enclosure, and we are calling this the flip method. So what this looks like in the field was attaching a clean enclosure to the top of the already pre-existing enclosure. And then we would rotate them in the water. So this makes for a water-to-water transfer of the smelt. So hopefully they don't get too stressed out. That's kind of, we were also kind of looking at that kind of stuff. And this was done every 2 weeks because in practice, this takes 2 boats and like 6 people and a lot of work. So we wanted to kind of test this all out. And there at the end, we just detach it, take off the top and put the lid back on, and we're out. So then our second question, we were talking about the 2 different sites. And this, we chose 2 sites that were pretty different habitats. And the first is what we're calling Montezuma. It's in Montezuma Slough, right by Belden's Landing, for those that are familiar, but it's pictured on the map. And then the second is Rio Vista, which is just south of the town of Rio Vista on the lower Sacramento River. And these 2 sites have been used for enclosure studies in the past, so we knew that they were feasible going into it because we didn't want to also just add that to the list of things that we were testing out. So at each of the sites, we set up a total of 4 enclosures, 2 for each treatment. We were limited by the number of enclosures that we have. So we were only able to get 2 replicates per, and then we weren't able to include a no biofouling treatment. Control would be the word. But we were able to hold fish at the FCCL and use those kind of as a baseline control fish to compare to. So the setup was that we did this experiment over a total of 6 weeks. So that flip method was done twice at weeks 2 and 4. And week 6 is when everything comes out. So you don't have to clean everything up that week. And the scrub was done every week throughout. And each enclosure got 70 Delta Smelt. So to assess whether the biofouling or the sites was having any effects on the condition of the Delta Smelt, we measured their survival, hepatosomatic index, condition factor, liver glycogen, critical thermal maximum, and diets. These data were collected at the end of the experiment by taking a final count for survival. Critical thermal maximum was measured in the field, so a subset was taken out of each enclosure and to a treatment tank area, and they did that. That was all done by the fish physiology lab. And then everything else, we just took a subsample out and preserved them appropriately to the method. Some things went into liquid nitrogen, others went to ice or formalin, that kind of thing. And so for all those metrics, we wanted to kind of try to make a prediction of what type of relationship we could expect to see with biofouling. But since all these metrics, including biofouling, are pretty multifaceted, we weren't able to come up with a single conclusion. So we kind of have to let the data talk for itself. Like, for example, the biofouling could reduce the water quality inside of the enclosure, as I was saying earlier, and that could lead to increased mortality of the smelt, or the macroinvertebrates could be increasing survival of the smelt inside. So we just had to test it out and see what happens. So we're going into the first set of data. Here I have the results for hepatosomatic index. For this data, we found that the control fish that were held at the FCCL in that orange box scored significantly higher than all of the fish that were in the field, but there was no significant effect of hepatosomatic index on the smelt by the biofouling treatment. So that would be between the exchange and scrub. So those two sets of box plots next to each other. So because there's so many plots to look at, that was a lot of different metrics. That's just one example of the data, and I'm just going to kind of show the results. All of the data for the metrics that we measured on the smelt, except for the diet, I'll get to that in a little bit. None of that was significant in what we found according to the biofouling. But it exited out with it. However, okay, when we look at these metrics by site, we did find some significant effects. Here's the data for the critical thermal maximum. And as before, like the FCCL control fish scored higher than all the other fish. But this data found that the fish held at Rio Vista scored significantly higher than at Montezuma. So this same pattern held for condition factor and liver glycogen. We did not find any significant effect of location on survival or condition factor. So our results indicate that some of the Delta Smelt condition metrics were significantly affected by site, but not by the biofouling. But since here's the diet data, and since there's so many factors to consider with diets, it's getting its own slide here. But we're still waiting on some other data and some more analyses to kind of get stats. But anecdotally, we can kind of tell a story that the species most commonly eaten at Montezuma was Limnothoina, and the species most commonly eaten at Rio Vista was Pseudodioptimus. Both of these species hang out in the water column, so we wouldn't really expect those to be coming out of the biofouling itself. So it's probably more attributed to the site rather than the biofouling. And then we also measured a bunch of different effects on the conditions inside of the enclosures. So this is kind of getting at whether the smelt are really experiencing something different being caused by biofouling, or if they care or not. So we measured the macroinvertebrate abundance, algal biomass, flow inside of the enclosures, and then collected zooplankton samples. So for macroinvertebrate abundance and algal biomass, we measured the amount of macroinvertebrates and algae that grew on the surface of the enclosure throughout the experiment. The flow inside of the enclosure was a relative measurement. We used what's called clod cards, and it's just this, as water flows over it, it kind of erodes away. And so we can get the weight. And so it's relative, and it tells you like between each enclosure if they're experiencing more or less exchange of water. And then we also collected zooplankton. This was done every 2 weeks using pumps from inside of the enclosures. So for these methods, they're a little more straightforward, and we were willing to venture some predictions. So for the macroinvertebrate abundance and algal biomass, we hypothesized that as biofouling increased, so too would those metrics. This also makes a lot of sense because those are the two main components that make up biofouling. And then inversely, we predicted that flow and zooplankton inside of the enclosures would decrease with biofouling, thinking the increasing growth on the enclosures would reduce water and zooplankton's ability to enter the enclosure. So here's some more data, and I have the results for the macroinvertebrate abundance being showed. And we found that the flip method significantly reduced the macroinvertebrates that were growing on the enclosures compared to the scrub method. However, we did not find a significant effect of those two treatments, so the flip or the exchange method or scrubbing method between algal biomass and flow inside of the enclosures. And then next, when we were looking at that same data by site, we found that the flow was the only metric significantly affected by site. This should likely be due to the larger tidal influence Montezuma experiences compared to Rio Vista. So the flow there is actually higher. So it kind of just follows what's being experienced at the site. But that was what we found. We found that that was the only significant effect by site. And then same as the diet data, the zooplankton is getting its own side because there's so many factors to it. And like the diet data, we are still like waiting on more data. So we want to compare this to other surveys that are in the area, so we can get kind of if our equipment has a bias or not, and we can kind of try to get to those answers. But with what we have now, we can kind of anecdotally get a little story. So we found, let's see. So we'll just look at what was most important in the diets for this quick conversation. And we found that Limnothoina was pretty dominant according to the count at Montezuma. And then Pseudodioptimus was not nearly as dominant as Limnothoina was, but it is one of the more abundant individual species at Rio Vista. And so to wrap this all up, we found that all the smelt condition factors that scored significantly, all the smelt condition factors that were significant scored higher at Rio Vista than at Montezuma. Macroinvertebrate abundance was significantly reduced by the flip method, but this was the only factor that was significantly affected by biofouling treatment. And anecdotally, we found that Pseudodioptimus was the most common food at Rio Vista and not at Montezuma, indicating maybe the food at Rio Vista was a little higher quality for Delta Smelt. And so we'll kind of looking into the future, since our results indicated that biofouling had limited to no effect on the condition of the smelt inside of the enclosures, we want to focus future enclosure work on investigating habitats and reduce our efforts on mitigating that enclosure biofouling. So with the freedom from reducing our efforts on biofouling treatment, we can utilize those resources to expand enclosure studies to new sites and new habitats. And then by expanding enclosure studies to more habitats, we can deepen our understanding of how Delta Smelt respond to different habitats and locations. And this can allow us to more effectively utilize enclosure studies to inform how things like summer fall habitat actions are affecting Delta Smelt. And with that, I want to thank everyone that made this possible. It was a huge undertaking. There's a lot of field work and a lot of lab work that was done. And I think I have time for questions.
Thanks for that talk. I was curious, I may have missed this, but did you do anything like not treat any of the cages? Like, were there some cages that you just didn't treat and look at that effect?
Yeah, we didn't have enough enclosures to be able to have replicates and do a no treatment, like no scrubbing, no exchange treatment. But we were kind of like, well, regardless, like we're going to go scrub those enclosures just to make ourselves feel better. So, so we felt like that's kind of will be the baseline regardless of if we did or did not do a treatment. Here's one.
I don't know if I missed it. Did you say when you had the cages in? What time of year?
Great question. We did this in like September through October. So 6 weeks total. So almost 2 months, but yeah, it was, yeah, pretty hot. And this was 2023. So also like that kind of Rio Vista Montezuma, it might've been a little switch just because it was such a wet year and the conditions were very different than what we would expect normally. I think that's it.
All right, thank you, everyone. Excellent fishes, all of them were wonderful. We're now going to go to lunch. We have a little extra time for lunch today, so be back here at 1:40. Any other announcements?
Speakers for the next session still need to get there.
Yeah, if you're speaking soon, get your slides uploaded. Ideally now and not at the end of lunch. All right, folks, we're going to be starting in 1 minute ago, so please filter in and take your seats. All right, everyone, we're starting. Okay, I know we're coming in after lunch. It's a little people are filtering in. Please quiet down. We're now going to our salmon session, and the chair of this session is Marcia, and I will let her introduce the first presenter.
Thank you. Good afternoon, everyone. My name is Marcia Scavoney-Tansey.
and welcome to IEP Workshop Section 14, Salmonid Habitat Monitoring and Modeling. Our first presenter is Alison Collins. Alison is a senior research specialist with the Metropolitan Water Resources of Southern California, working with salmonid ecology. Her work is focused on utilizing science to inform salmonid-related management and policy section. Welcome, Alison.
Thank you. Thanks everyone for coming back to lunch. Kind of as this project is trying to reorient ourselves in the processes we work in, I want to just take a moment to acknowledge that we're on the ancestral territory of the Miwok and Nisenan people and affirm their sovereign rights as First People. So today I'm going to speak about quantitative tools and frameworks that can be used to help a group of people work collaboratively in a transparent process to achieve salmon recovery. As this graphic from British Columbia shows, there's a lot of people that care about salmon. There's a lot of people that are implementing different actions to help salmon, to recover salmon, ideas and values around salmon. And there's a lot of resources that are dedicated to this species. It's not easy, though, to balance the limited natural resources that we have among the many different parties that live in and use the Central Valley. We have a lot of competing objectives that we're trying to manage at different scales across different landscapes in a really complex ecosystem. For example, what happens when one group comes in and says that they want to recover salmon because they want to have more fishing opportunities. And one way they think they can achieve that is by increasing hatchery production. And then you have another group that comes in and says, we want to help salmon by increasing their survival. And we actually have this great idea that we can flood rice fields as a means to increase growth and then promote overall survival. And maybe your groups talk about this and they say, like, that's a great idea. And we actually think that flooding rice fields might lead to more fishing opportunities and that our idea of increasing hatchery production might increase survival. And now you have a third group that comes in and says that we actually really care about seeing natural origin fish in the system. And we're concerned that increasing hatchery production might reduce the number of natural origin fish, but flooding rice fields might increase this number of fish. So what we have here is a lot of competing objectives, a lot of different interests that are in conflict with one another. As you keep adding objectives to this tables and alternatives and how to achieve those objectives, your table is going to grow exponentially, right? But luckily, there's quantitative tools and frameworks that can be used to, solve this problem in a transparent and collaborative way, which brings us to the process that I'm presenting on today. The goal of the Reorienting to Recovery project is to identify preferred broadly supported scenarios across all interested parties that support recovery of Central Valley salmonids. So this work is going to identify a suite of implementable and impactful actions to advance recovery of the 4 distinct runs of salmon in the Central Valley, while establishing broad support and buy-in for these preferred actions. We're accomplishing this by using decision support tools and a structured decision-making framework. This group and process will be producing recommendations, not formal decisions. However, it is anticipated that these recommendations will inform future decisions around coordinated actions to recover salmonids that can be implemented by a variety of parties. So as I mentioned, this project is using structured decision-making to create scenarios to recover salmonids while balancing multiple other competing objectives. So structured decision-making is a systematic, transparent method that has been effectively used for navigating many complex environmental resource issues. It helps groups work collaboratively to build a shared understanding of the trade-offs and make informed and transparent choices. As a planning framework, it consists of a set of core principles, a series of fundamental steps, and a series of applied methods and tools that draw from decision sciences. So it really allows groups to come together and talk about what's the context of what we're deciding about, what kind of objectives do we want to achieve, what alternatives can we implement, what are the consequences of implementing those alternatives, what are the trade-offs that exist once you implement those actions, and then once you kind of set on a group of actions or a strategy put in place, you can monitor and learn and adjust. And while I mentioned that this is really great for large, complex natural resource problems, Corey and I have used it many times in our marriage, including if we should remodel or buy a new house and which car seat to buy for our children. So it can be used in lots of different places whenever you're trying to make a decision. So in our process, the first step was to define what do we mean when we say we want to recover salmon. So we gathered a group of experts on salmon biology, and over several meetings, this group developed measurable objectives that are based on the viable salmon population parameters. So those include abundance, productivity, spatial structure, and diversity. The group also identified performance metrics as ways to measure the objectives and set specific targets. So for example, here is productivity. We have a measure of cohort replacement rate, and there's specific targets that we want to meet in this recovery process. The group also decided that we're talking about broad scale recovery. So this is a recovery that goes like above and beyond delisting recovery criteria for these species, and it applies to all runs of salmonids, the listed and non-listed species. Right. So we're talking when people got together, like we want to see, be able to walk across salmon's back and have so many extra salmon that the killer whales can eat it. And we can fish without, quotas. Like we're talking about really big recovery here. And it's outside of the regulatory environment that some of the other recovery definitions are currently in. Phase 2, we brought together interested parties to develop a project catalog. So what are people doing to help benefit salmon? And define ecological, social, cultural, and economic values. So we had representatives from state and federal agencies, NGOs, tribal governments, water and ag interests all come together, and over a series of meetings, they told us what projects they were working on. And then we did kind of a storytelling workshop where we asked participants to tell us their values related to salmon by asking questions like, how do you imagine salmon recovery might benefit or impact you? And kind of collected everyone's ideas around that. We collected over 500 statements, and we distilled those into 24 objectives that we could track throughout this process. So an example of this is values we heard were traditional food source, like salmon as a traditional food source, and recreational fishing opportunities. So we kind of grouped these values together into a refined value around salmon harvest, and then developed some performance metrics that we can track throughout the system. So as we're kind of going along and evaluating strategies to recover salmonids, we can make sure that we're capturing all the values from interested parties that we heard. Phase 3, which we're in now, uses structured decision-making to collectively build and assess different combinations of management actions or recovery scenarios towards a broadly supported strategy. So we are using decision support tools to predict the consequences of different recovery actions and scenarios on the objectives defined in Phase 1 and Phase 2. So this serves several purposes, such as improving our understanding of the effects regarding scenarios on the objectives and discussing the trade-offs. So the Reorienting to Recovery, or the R2R decision support model, is adapted from the CVPIA Science Integration Teams model. That's an open source model. So our modeling team, essentially Flow West, took that model and made extensive updates to better represent hatchery dynamics, adult harvest rates to better reflect current harvest numbers, and in the San Juan Key Basin, they extended spring run habitat up to Friant Dam. So recovery actions that can be modeled in this decision support tool are related to the 4 H's: habitat, hydrology, hatcheries, and harvest. Examples of some of the actions we can model and represent are like gravel augmentation projects, reintroduction, restoration, changes in flow, the timing and magnitude, changes to hatchery and harvest practices. There's a lot of different knobs that we can kind of turn and tweak. So once the group decides what kind of recovery scenarios they want to evaluate, those can be ran through the decision support model, and results are presented in a consequence table. So at this point, our process has done 2 rounds of modeling. We're about to embark on a 3rd round. I'm going to show you generic results today just to show you how consequences tables work, and they can be used to kind of evaluate trade-offs that exist. So this is what a consequence table look like. On the left, we have the objectives. So remember, those represent the biological and non-biological values, the interests that were defined in Phase 1 and 2 of this process. In yellow, the columns on the top right represent the different recovery scenarios. So each column represents distinct scenarios that contain different actions to recover the species. And on the right are the predicted consequences of recovery actions on each objective. So the legend at the top shows you how to read the different colors. And just because talks are so short, I'm just going to say that at the moment, darker colors of blue and purple are more preferred. All right, so this is I'm going to show you some real results, but in a generic way to kind of just talk through this consequence table. So I selected some biological metrics related to adult abundance, cohort replacement rate, and PHOS. And then we have some other socioeconomic values that were identified, including ecosystem health, wetlands, ocean harvest, water supply, flood risks, and hydropower. So you don't, if you're not an expert in salmon biology, it's okay. You don't need to really know what these means for the purposes of this particular talk. The performance measures are how we're going to measure each of these objectives, and the preferred direction just reminds you if we want to see higher or lower values predicted in the consequence table. This is showing results for two alternatives. Again, I'm not giving you any details about what are in these alternatives. It's just for illustrative purposes to show you kind of the differences that exist here. So what I see when I look at this table, remember that colors in dark blue or purple are more preferred. So I see that alternative one, kind of looking down this alternative, it's doing a better job at achieving values that we prefer related to the biological metrics and ecosystem health, shown by these dark colors here. And it's not doing as good of a job satisfying some of the other objectives that we care about. And the opposite is true for alternative two, right? So like looking at adult abundance in alternative one, we have a lot more fish that are produced under this alternative in the Sacramento and San Joaquin systems. That also directly relates to marine-derived nutrients, which is a proxy for ecosystem health. This value includes the number of fish that are spawning, right? So it makes sense that if you have more fish present at spawning grounds, you're going to have a higher value of marine-derived nutrients. Alternative one produces less fish that are available for ocean harvest and has less water supplies. You can look down this table and look at the objectives that you care about. So this is just to give you an example of some of the trade-offs that exist between a couple of the alternatives that we've looked at. And you can imagine that this is going to lead to like a healthy discussion along participants about what trade-offs there are, what scenarios they prefer, and how they want to prioritize these objectives. So we are actually having that discussion with our decision makers next week during a 2-day workshop. Again, these decision makers represent state and federal agencies, NGOs, water and ag interests. We're working in a separate process with tribal governments to get their input and feedback and preferences in this. And at this workshop, we'll be asking participants to brainstorm the next round of modeling scenarios that we want to do to try to balance some of the trade-offs that are existing in the results that we're seeing. I hope that today, I've shown you that there are quantitative tools and frameworks that are in place to help groups with diverse values and interests explore complex natural resource problems with competing objectives. These tools really do allow a diverse group of users to come together and in a collaborative process to build an understanding of what they're trying to achieve and examine the consequences of implementing different alternative recovery actions and discuss the consequences of what implementing those actions are. So tools like this really allow diverse groups to make well-informed, transparent choices. With that, if you're interested in this project, there's a whole slew of resources. Also just want to mention that I'm one of a 20-person planning team. So all of us are happy to talk to anyone who's more interested. And then I want to thank our funders, the Delta Science Program and state water contractors. So with that, I'll take any questions.
Hi, great talk.
Thanks.
I was just curious about the interpretation of those numbers that you just had in your last figure. For example, where the alternative one had 6 million adult salmon, why are there only 500,000 that could be harvested? Is that just regulation? It wasn't quite clear to me why that would occur.
Yeah, it has to do with different assumptions that are in the alternative regarding harvest and hatchery practices. So those different assumptions and different alternatives lead to different outcomes on harvestable fish. Okay, thanks, you guys.
Thank you, Alison. Okay, the next presenter is Brian Matias. Brian is a supervisory fish biologist with the U.S. Fish and Wildlife Service in Lodi. He leads studies to assess the survival, migration, and abundance patterns of juvenile Chinook salmon and still had migrating through the Central Valley. Welcome, Brian.
Thank you. So today I'm going to talk about the estimating abundance of endangered winter run Chinook salmon exiting the Delta. And this is a project that my group has been working on for several years with a bunch of collaborators. And I want to specifically call out Russ Perry. He's been doing the vast majority of the analysis on this project. And this project is adapted from several that he has given in the past couple of years. So several years ago, there was a group of individuals that was looking at the winter run monitoring network. And they came to the conclusion that we didn't need more monitoring for winter run Chinook. We just needed more focused monitoring. And they came up with 6 system-wide recommendations. The first being making sure we were incorporating genetic run identification, getting abundance, looking at survival, diversity, condition, and data access. This project is really looking at the first 2 components of that. So the genetic run identification and abundance components. So we did this project for both the Sacramento trawl site and the Chips Island trawl site from DJF&P. And today I am going to be focusing just on the Chips Island component of this effort. So why is abundance at Chips Island important? Well, it is a critical nexus between the freshwater and ocean phases for juvenile Chinook salmon. It is a really good place to monitor the status of endangered populations because it can provide an early indicator of year class strength and allows us to take action if there is low abundance. This is also really useful to inform life cycle models. So the science integration team winter run decision support models ideally will be calibrated with abundance data, calibrated and validated with abundance data. And this is one source of information that we can use to do that. However, there are plenty of challenges that we have when we're sampling at Chips Island. One is just the mass amount of water that's passing past Chips Island every day and the small fraction of water that we are sampling. So if you squint really hard, you can see a little white dot up there, and that is actually one of our trawls. So this really shows you how big of a needle in the haystack we're actually doing when we're sampling. So that is one of the challenges. The other challenge when we actually catch a fish, what are we looking at? Are we looking at a hatchery origin fish? If so, what hatchery did it come from? And the best tool we have for that is coded wire tags. However, there are certain runs that only have the 25% mark rate. So not all of our hatchery fish are actually marked with coded wire tags. If it's a naturally produced fish, what population did it come from? Did it come from fall or late fall, which are harvestable? Did it come from threatened spring run or endangered winter run? And the best tool we have for that is genetic stock identification. So this study started off as a 5-year, pilot study, and it can really be broken up into 3 different parts. Before I get into that, I want to talk a little bit about this figure. So we've got a couple of different boxes. We've got the boxes with a nice bold blue border, and that is part of our existing monitoring network. The boxes that have the dash border, those are the data that we are collecting each and every day we go out sampling. The circles, those are the model estimates, and ultimately we want to link that back and get estimates for run specific abundance. So linking that back to the little yellow box on top. Now, there are 2 parts that we are modifying with this study, and those are the boxes in green that I'll talk about in a little bit. So the first one, we are pairing coded wire tag releases with acoustic telemetry. So we are taking a small proportion of the hatchery production with acoustic tags and releasing them with the coded wire tagged fish. This way we can use acoustic telemetry then to estimate the survival to both Sacramento and Chips Island. So how many of our coded wire tagged fish are expected to make it to the trawl sites? It also gives us information on run timing. So when do they actually get to these trawl sites? Part 2 is linking our coded wire tag catch and ultimately estimating our trawl efficiency. So traditionally, our sampling at both of these sites is done 3 days per week. With our efficiency sampling, we're increasing that. So on average, we're going out roughly 5 days per week between December and May. And this allows us to maximize the number of coded wire tagged fish we are catching. So then to estimate our trawl efficiency, we have known coded wire tag catches. So we know how many coded wire tags we're catching at any given time. And we have estimates of the number of coated wire tagged fish available to be captured at each trial site from those paired releases. And then the genetic component, we're again linking our trial catch with genetic IDs, relying on the increased sampling. So sampling again at 5 days per week, so we can maximize the number of true genetic winter run that we're catching. So then we can estimate our winter run abundance with that genetic sampling. We know the proportion of fish that are actually true winter run in our sampling or in our catches. And then we are able to use those efficiency estimates that we talked about in part 2 to scale our winter run abundance to get the true winter run abundance. Okay. Part 1, just quickly going over some of the paired release and acoustic tag summaries. So with this initial part of the model, we have data from 2016 to 2021 and 31 different paired releases. These come from across the years. We have, I think, at least 4 paired releases every year, and they're coming from the various hatcheries and runs. So we have representatives from each of the different runs and numerous hatcheries. All in all, we tagged close to 10,000 acoustic or 10,000 fish with acoustic tags, and roughly 1,500 of them were detected at Chips Island. At the same time, with these paired releases, there were approximately 12.5 million coated wire tags released, and just shy of 1,900 were caught at Chips Island. On to our fun coated wire tag catch and trawl efficiency. So here we've got our base model efficiency. So to orient you to this figure, each of those little dots represents the individual trawl efficiency for a paired release group. Now we've got this oriented based on the run and hatchery. So on the left, we've got Coleman Fall. Next to that, we've got Coleman Lightfall, and so on. The red line that's going across is our overall mean efficiency. So on average, we're about 0.75%. So this is actually remarkably consistent across time and across all of the releases. Individual release groups are going from maybe about 0.25% all the way up to maybe 1.75%. So I would argue we're pretty efficient, or we're doing a good job at estimating our efficiency. We also integrated or looked at factors affecting trawl efficiency. So some of the fixed effects, delta outflow, velocity deviation representing tidal movements, interaction between outflow and velocity, tow location. We tow in 3 different locations: north channel, center channel, south channel, and then tow direction, whether we're going upstream or downstream. The 2 biggest ones, delta outflow and tow location. So we are less effective at, or less efficient at catching fish at high water levels. And the south channel, for whatever reason, has the highest efficiency. Okay. Getting on to some of the genetics data. Here we've got the data for the genetic samples, and each of these are broken up into the length at date category. So we've got the genetic samples that we collected and analyzed kind of on the left in that yellow box. And on the right, it's the assignment for actual true winter run. So if we take a look at 2017 for the genetic samples analyzed, we looked at 17 late fall length at date fish. For fall run, we looked at 24 and so on. All in all, we collected about 3,000 genetic samples throughout these years. 212 of them were assigned to winter run, and roughly 95% of the true winter run were within that winter run length at date group, and a couple of them were in the spring run, and a pair were in the late fall run. Okay, on to the fun stuff, the actual abundance estimates. So here we have our abundance estimates for genetic winter run from 2017 to 2021. Ranges, we had a low in 2021 of about 32,000 and a high in 2020 of about 92,000. So taking a kind of a step farther, Russ Perry has been doing a few more analyses on this. And I was kind of interested to see how well we were doing compared to other estimates of abundance. So over a decade ago, Brian Piper published a manuscript looking at genetic winter run abundance at Chips Island. And We're also just interested in applying the trawl efficiency model to the length at date winter run. So how well do those two methods match? So here we have winter run abundance estimates from 2008 to 2021. Again, we've got those three different methods. The weird pink color is from Piper et al. 2013. The black is trawl efficiency, and then the length at date is the weird brownish orange color. So again, this is the length at date model applied for the trawl efficiency model. And the first thing that I noticed is looking at the Piper et al. Data and the trawl efficiency model, they're very, very close. Especially in the first 3 years. The last year, 2011, there was a difference or it was a high water year. And the Piper model didn't account for the high water as well as the trawl efficiency model does. Length at date winter run, big surprise. We are overestimating abundance using the length at date model. That is not a huge surprise by anyone. However, I do want to point out 2017, also another big water year. For whatever reason that year, there were a lot of fall run fish that were collected in, or that were classified in that winter run length at date category. So it was just fall run were everywhere that year. Kind of moving on to some of the future efforts. So we are continuing this project and we have transitioned to long-term operations. So with the goal to develop an annual abundance estimates for winter run, both entering and exiting the Delta. Again, I didn't talk at all today about the efforts at the Sacramento trawl site. And we did all of the paired releases for winter run in 2021 and 2022, and we're still analyzing those data to get these estimates. And we've got funded efforts to apply these models to estimate both spring run abundance and fall run abundance. So with that, I really need to thank all of our partners and our funding agencies and the copious amounts of field staff that have helped throughout this project. And I think I've got a couple of minutes for questions as well.
Yeah, great talk. It seemed like in your efficiency analysis that the channels were significantly different. I just wonder if you might comment on why and what might be going on there.
We've had that conversation. It was a number of years ago now, and I don't remember all of the specifics. I think it has to do with channel morphology. That I think it was a south channel that was a little bit higher efficiency, and I think it, for whatever reason, the flows might be pushing them a little bit more towards that south channel as they're coming out of the Sacramento River. That's our best working hypothesis at this point. All right, thank you very much.
Next one. I'm sorry. So, thank you, Brian. The next presenter is Matt Peterson. Matt is a senior fisheries biologist with Fish Bio in Chico. His recent work focuses on steelhead predator-prey dynamics and mark recapture modeling to estimate abundance and movement patterns on both south and native and non-native species. Welcome, Matt.
All right, thank you, everyone. Well, today I'm going to be talking about an exciting new project we're getting up off the ground. We began this work in late 2023 and using multi-state mark recapture models to both estimate mortality sources of immigrating salmonids and also assess how well restoration activities might be working or different restoration areas. First, I'd like to thank our funding sources and my co-authors. This project wouldn't be possible without the efforts of Dr. Reed Nelson and Elizabeth Greenheck, both from George Mason University. And I'd also like to acknowledge some feedback we've received on the project thus far from various agency staff, especially on the one of the objectives I'll talk about today. So this project is essentially doing our homework before we do the real final exam. And I am treating the final exam as conducting a very large, complicated acoustic telemetry study that may cost millions of dollars to do. So basically this project is essentially figuring out what types of questions we can ask with these multi-state models, and whether we can partition mortality of salmonids as they migrate downstream through any system in the Central Valley watershed. And then ask also, can we assess the survival benefits of habitat restoration with these same multi-state models? We think we can. We think we have some good ideas, but one of the objectives is to specifically reach out to folks who have done these types of studies, that are actually working on habitat restoration, whether it be in the upper river, in the delta, or in more tidal habitats down in the estuary. And so we're trying to incorporate some of that feedback into our models, our different scenarios, and these different research questions. And then eventually down the road in several years, which I'll get to in a second, we hope to share those findings in several publications and also share the source code to allow other folks to be able to use these models to ask specific questions that they are interested in using what we've learned in our assessment. So we're not doing the fun field work yet. We're doing the office work. We're using a simulation-based approach to answer these questions so that it's relatively cheap to do. And we can make all the mistakes we want, right? This is more of a personal stance. Like, I think it's really important to know whether your question can be answered by the study design and study approach you're using. And so this is one of those ways we can do that. So we are fairly early on in this process. Right now, we're developing our initial research questions, our initial models, and incorporating feedback on both the mortality side of things and the habitat restoration side of things. This year and next, we're going to be basically running all those models and with those different questions. And one thing we really want to take advantage of is that there's a lot of new tag technologies, acoustic tag technologies, that can both sense temperature, sense pressure, and also new ones that can also detect whether a fish has been consumed by another fish predator. But when we're doing all this, we want to make sure that we're getting reliable, robust results from these types of studies. So that the technical, this is as technical as I'll get today, because I'm really out over my skis on this stuff. This is more Dr. Nelson's expertise here. But basically, we are taking the Cormack-Jolly-Sieber models to the next step by using multi-state models. So for Cormack-Jolly-Sieber models, you essentially only can estimate two different states of the fish. They're either alive or they're dead. That's fine. We've learned a lot of good information about how salmonids are surviving through the system by using Cormack-Jolly-Sieber models. But we think the multi-state models can add more information to what we've already learned. And the reason why is because we can both estimate survival and as long as you develop your questions, develop your research design or your study design the right way, and collect observable data on different states, you can then ask more nuanced questions about why fish went in certain habitats. Did it go into habitat A or habitat B? Or if they died, how they died. And so a recent example is from the East Coast. This is work published earlier this year by Minsinger et al. And this is a study where they used such a multi-state model to partition mortality of Atlantic salmon smolts as they moved down, and I'm blanking on the river right now, as they moved down the river. And what is really important to highlight is that the reason why the fish died changed as they moved downstream. It's not surprising, right? But in this study, they had a lot of unknown mortality, a lot of fish predation, mortality in the upper river, and then a lot of that transitioned to mammalian mortality down in the estuary. And so one can easily imagine that same type of scenario occurring in the Central Valley. So we think this has tremendous promise here. So transitioning just to one of our potential research questions or one of our scenarios. Here we have just an example conceptual model where we've released acoustically tagged fish with predation detection tags in the upper reach, the reach named labeled B. Those fish then migrate downstream. They're either detected by receivers or not, and those receivers are denoted by the black dots. And then we've set up reaches where they die at certain rates and they die from certain causes. And so for reach 1, we have poor detection probability in reach 1, and they die from an unknown cause. We can't quite figure that out. In reach 3, we have good detection, and those fish are lost from fish predation. And so what we're trying to get at with a simulation is to figure out under what scenarios, so if you release lots of tags or very few tags, how well does this model work in this particular scenario? And as you can see, and it's not really a surprise, the more tags you release, the better the precision of your estimates. So the dots on the graphs represent the truth, essentially, of how those fish died. And then the box plots show how, based on like 100, I believe it's 100 simulations, how far apart your simulation is to the truth. So you can see a lot of variability in the top row where you've released very few tags, and very good precision on the bottom row where you've released a lot of tags. So what we want to try to do is find the sweet spot here, right? Because you might not have the money to estimate lots of different states like we have here, or you might not have enough money to release 1,000 tags. It might cost $800 a pop or $500 a pop. I think that's all I was going to say on that. So that covers the predation mortality and trying to partition the mortality sources. The other component of this project is to try to assess the survival benefits of whether fish that use different habitats, and particularly restored habitats. We think this has a lot of value given the high amounts of restoration that are proposed in the Central Valley through programs such as California Eco Restore and the Healthy Rivers and Landscapes programs. We think just getting the data on how tagged salmonids are using some of these restoration area has just value there to learn how they're using the restored areas. And for this particular component, we're actively seeking researchers and managers and restoration practitioners to give us feedback on some of the questions they might want to ask of their particular restoration areas. So like I said, project is just getting underway. We are refining all the different questions and scenarios and the models, incorporating feedback on objective number 3, which is the habitat restoration component, and obviously continuing our simulation-based approaches. That was a really quick rundown of a very complex project. I highly recommend everyone to check out our project fact sheet, which gives a little bit more detail on this project. And if anyone has questions, feedback, suggestions, feel free to ask now, or please email me or give me a call. With that, thank you.
Thank you, Mike. Any question?
Thanks, Matt. Awesome talk. I was just curious, how do you get at the causal differences of mortality? How do you know whether a bird or a fish or a mammal is eaten?
That is a good question. For the fish predation, it's fairly simple with the newer predation detection tags, assuming those are working good. To pin down the mammalian or avian predation, I know Mensinger et al., they used the temperature sensing tags, and they were able to get that information off those tags and essentially figure out whether the tag was in a bird or a mammal based on the internal temperature of the tag. Thank you. Good question.
No more questions?
All right.
Thank you, Matt.
All right. Thank you.
Thank you so much. Our next presenter is Garfield Kwan. Garfield is currently a Delta Science Fellow and postdoctoral research at UC Davis. He is working with metabolic index models to determine the viability of Chinook salmon and Delta. And the last but not least presenter is Brandon Lehman. Brandon is a researcher with the UC Santa Cruz Institute of Marine Sciences, and he's affiliated with the NOAA Southwest Fisheries Science Center. He worked with a variety of anadromous fish across California with a focus on how anthropogenic stressors affect interaction between predators and prey. Welcome, Brandon.
Thank you. Thanks for the introduction and thanks for the opportunity to speak here. I'm going to be sharing some work about artificial nighttime illumination, which is a topic I don't think has received a lot of attention in our system. So, throughout the history of life on Earth, animals adapted to reliable cycles of night and day, using the cover of darkness to evade predation, shifts in the zenith of the sun to navigate, and the timing of sunset and sunrise to cue migration and reproduction. Artificial light is a form of pollution. It's an environmental disturbance that affects organisms and the way they interact with their surroundings and with each other. There's been a recent increase in attention in the scientific literature to the effects of artificial light in wildlife, although most of this research has focused on birds, insects, sea turtles, and mammals. There have been a handful of studies on fish, and a lot of that work's actually been done in the Pacific Northwest and on salmonids, which is fortunate for us. But there's been a fairly limited conversation about the effects of artificial illumination on fish in California. That being said, there's a good reason to think about light pollution here in the Bay Area. This is the San Francisco Bay, and this is the Delta here. This is this little, oh good, you can see that. That's Rio Vista right there. We've created a huge infrastructure of nighttime lighting. In 2018, UC Santa Cruz was funded by a CVPIA Fish and Wildlife Restoration Grant to study how physical habitat features influence predation on juvenile salmon in the Central Valley. The intention of this was to collect data that allows the CVPIA science integration team to parameterize a structured decision-making model that helps prioritize funds that are allocated for restoration projects. So essentially our goal was to help figure out how many fewer salmon die if we mitigate for the effects of specific types of habitat features that are present in the Delta. One of these turned out to be artificial illumination. We started with a literature review that helped inform subsequent field experiments, quantifying the predation risk of juvenile salmon around artificial lights in the Delta. And the summary is pretty simple. Light pollution is bad for juvenile prey-sized fish, such as salmon smolts. Next, we conducted a series of field experiments, which I'm not even going to be covering the methods of those there, but basically we were able to, we observed extremely elevated levels of predation on juvenile salmon in areas that were artificially lit at night in the Delta, which allowed us to develop a model for predation risk in relation to specific illumination levels in these lit areas. So the next task would be to translate this to a landscape or population level. However, there's no inventory of lights in the Delta. And in fact, there's not really any inventory of lights hardly anywhere because there's not a lot of regulatory framework about installing lights. You're able to install lights generally anywhere, anytime if you want. And there's not a lot of rules about that. So we wanted to inventory all of the lighting sources in the Delta, and we started off thinking about using satellite imagery or flying drones around at night with cameras. But in the end, these were kind of boondoggle ventures, and we ended up collecting this data through brute force. So we essentially drove around the Delta at night in a little aluminum skiff. This is a light meter. They're cheap. They're easy to use. This thing costs about $2,000. And we mounted it to the bow of our boat and waited until well after sunset on moonless nights and drove around with a GPS. Anytime we got to an area where we observed illumination that appeared to be shining on the water, we'd stop and take transects to develop two-dimensional raster data of the distribution and intensity of light. So this is what that looked like at a landscape level. In general, the Delta is a pretty dark place because it's not super urbanized. Although we did identify about 150 areas that had measurable amounts of artificial illumination, docks, bridges, marinas, streetlights, houseboats that were permanently moored, et cetera. And these light sources are generally evenly distributed through all of the channels that are relevant for migrating fish. So up the San Joaquin, down the Sacramento, and all the steamboat, Sutter, Slough, Georgiana, et cetera. There's a lot of variability in the brightness of these spots and also the area. Like some of these are just one single light source. Others could be an entire like marina complex. So I'll show you a couple examples of what that data look like. This is the Old River Railroad Bridge, just downstream of the South Delta intake pump facilities. So any fish that's getting sucked that way from the North Delta is going under this bridge. And you can see they're super bright lights. The channel here is like maybe a couple 100 feet wide and it's super shallow there. So any fish that's headed this direction is gonna be caught up in that. And as far as I can tell, these lights aren't really even serving any kind of purpose. Here's another example. This is some industrial facility along the Sacramento River near Isleton. The light here only so this is where the picture is taken here, and the light is only permeating at least a measurable amount about halfway across the channel. And again, these lights are not intending to light up the water for any reason. They're just in the parking lot. This is the Rio Vista Bridge, which is kind of a medium level of brightness all the way across. So any fish headed this direction is going to get caught up in that. So yeah, we have this for basically all of the navigable waterways in the Delta now. And so this, what I'm going to share here is kind of more of a thought experiment than anything else. This isn't really like, the numbers aren't really quotable. And I guess this is like my comic relief slide with all the equations that I can't really fully explain right now. And I also have a fee, but it's a totally different fee than Garfield is explaining there. But basically, we know from telemetry studies that fish migrating through the Delta, and we're going to use the example of the main stem Sacramento from like Freeport to Rio Vista, they can experience anywhere from 100% to zero mortality. So like if 1 million fish enter the Delta, depending on the conditions, when these smolts are migrating through, like all of them might die if it's a really poor conditions. Sometimes almost all of them make it. It's super variable across years. So we have that whole like suite of baseline survival scenarios. And what we did is we broke that 50-kilometer section of river into 10-meter segments, assigned a proportional survival associated with whatever the baseline survival rate is. And then along these transect lines, like let's use this one as an example, these are a bunch of points. So each one of those points has a baseline survival rate, and then we assign the additional risk associated with the actual brightness of that point. And that's data that's informed from our empirical studies back in 2019. We take the average of all of the points along any given transect to come up with a new adjusted survival for that 10-meter reach. And then if you multiply those all together, you get an adjusted region-wide survival rate for an entire cohort of fish that are traveling through. And then if you subtract that from whatever your baseline scenario was, you have an estimate of how many fish are dying because of these lights, or conversely, if you were to remove the lights and do that restoration action, like how many might it save? And again, this is super back of the envelope, but I think it's really useful to think about. And so this is what that looks like. On the x-axis, this is the baseline survival rate. So 0 fish are making it as a baseline, 100 fish are making it as a baseline, like not accounting for the lights. And then we'll talk about this black axis first. This is the number of fish that would be saved by removing the lights for every 1 million fish that enter the Delta. So like just in the middle of our baseline here, like let's say we're assuming a 50% survival rate, 1 million fish enter the Delta, 500,000 are making it past Rio Vista. If we were to turn off all the lights that we observed in our survey, we estimate that like somewhere between 200 or 300 fish would be saved, which doesn't sound like a lot, but as a percentage of the population increase for this like life stage, which is only a couple days long, and we're talking about fifths or tenths of a percentage point, which like sounds puny, but there's millions and millions of fish that move through the system every year, and it's year after year. And we throw all kinds of money at restoration action. We don't even know if it's having an effect on, a positive effect on whatever it is we're trying to measure. So anyway, it's just kind of something to think about. This is another product that came of this. It would maybe be a little ambitious to go address every single source of artificial illumination in the entire region, but can we rank them or prioritize potential restoration sites? And so at least using our framework for what might be affecting fish, each one of these points is a specific location, like it might be a marina or a bridge or a dock or streetlight or whatever. And then the darkness of that spot is a z-scored estimate of how many fish it might be affecting negatively. And there's some pretty clear outliers. And then on the axes, there's the surface area as well as the actual brightness intensity. And so in our framework, at least, it's not just one of those that is skewing all of these points. It also depends on the channel size and things like that. So that's just another product to come of this. So this is my summary slide here. I'm making a pitch to restore darkness. I think this is a worthwhile thing to think about in an ever brightening world. There's some really obvious solutions. Most of the time we have lights that aren't even doing anything at night, like parking lots and industrial facilities just leave lights on. And like sometimes it's for security reasons, but like who knows how much that really matters. And there's, you know, automatic timers. You can just ask people to turn them off when they're not using them. You could install scaffolding to just minimize how much the light is reaching the water surface. There's obvious benefits. Like I only talked about this one paradigm of juvenile salmon smolts swimming down this linear dimension of the river, but like this could have benefits to all kinds of other wildlife. It's presumably affecting all kinds of fish up and down the food web, as well as like non-aquatic species. And then it's also uncontroversial compared to a lot of other types of restoration actions that we talk about. It's inexpensive, it might even save people money, and it can be implemented quickly. And then the benefits reoccur, like you only have to do this once and then you're good. Like you don't have to go do it as long as it was left if they're leaving the lights off. And then of course the caveats is that lights are getting cheaper and we're installing them quicker than ever. And since we did this survey a couple years ago, going back out to these places, there's even more like housing developments with super bright lights that weren't even there. And then most importantly, this is obviously not a pitch to say this is a silver bullet solution to restoring salmon populations at all, but something worth thinking about. And with that, I'd like to say thanks, and I think I might have time for a question or two.
Yeah, really interesting ideas. Do you know anything about the characteristics of the light underwater, below the surface? In other words, what the fish might actually be experiencing?
Yeah, totally. Yeah, simplified or glossed by an entire body of literature and also studies we've done about obviously the light attenuates quickly underwater. And the way fish experience it depends on the angle that it's coming in and the wavelength or the distribution of different wavelengths that it's coming in. And that's like a huge question. But yeah, the risk estimates that we made were for the surface intensity in the delta. So in this actual environment. And so it's most comparable to just think about like, what is the intensity of the light at the surface of the water, assuming that you're not talking about a super deep habitat.
Yeah, I guess I get the sense that it's a logical place to start, but I could imagine that some of these lights could differ considerably, depending on the angle of incidence, perhaps more than anything. That's going to have a big effect on how much of those, what proportion of that light energy is actually penetrating the surface. Yeah, I think there's lots to do, obviously, but it's a great start. It's super interesting. Thanks.
Thank you.
Oh.
All right, thank you very much, everybody. We're going to go to a break now before our final session for the workshop, which is on troubled waters. So you're definitely going to want to stick around for that. And please be back here at 3:15. There should still be coffee in the lobby, so get your last little caffeine fix. And thanks again to ICF for sponsoring the coffee. Hi, everyone. I'm going to be moderating for you all. Thanks for sticking around for the last session, but please also stick around for the early career awards, which are going to happen right after this. So first up, we have Anna Furick. She's a PhD student at UC Davis. She's going to present on identification of toxic contaminants in California sediments using non-targeted analysis.
and Hyala azteca toxicity. So take it away.
Thank you very much. Also, you killed it on the pronunciation of everything. Hi, I'm Anke, and as you can see, my project is on non-target analysis. But first, before I introduce exactly what that is, I want to focus on the bigger picture, which you're all very aware why we monitor aquatic environments, but I want to talk about it in context of contamination. So monitoring aquatic environments ensures ecosystem health and organism survival, specifically in a world of increasing contamination. California in particular, where all these lovely photos were taken from various water bodies across the state, has several monitoring programs, such as my collaborator's sediment monitoring program, which use targeted analysis to focus on compounds that are already known to be toxic. But as people who study aquatic environments, we know that this isn't the whole story, and that there are plenty of chemicals out there that are unmonitored, things like emerging contaminants, transformation products, and complete unknowns like the 6-PPD quinone that was recently discovered that's killing the coho salmon up in Washington. So for emerging contaminants such as, 6-PPD quinone, how do we develop a cost-effective method for monitoring all contaminants that may be present in our aquatic environments? One approach is non-target analysis. Now, first, I want to explain target analysis. So this is when you use a chemical standard to identify a compound in a sample and quantify it. So if you think of just chemicals in a sample as like a sea, and each individual chemical is a fish. Targeted analysis is like a fishing rod, where each bait is associated with one chemical. This means you have a limited scope because you're limited by the number of chemicals you have. But you get instant identification because you know exactly what your bait is and you match it to your fish. Or in this case, you know exactly what your standard is and you match it to your chemical. Unfortunately, we have a much bigger problem than just the limited scope we're working with. So the parallel approach is non-target analysis, which is theoretically capable of catching all chemicals that might be in a sample. Think of this instead of a fishing line, it's like a net. So this is a broad scope approach that you're able to scoop up everything that might be in your sample. But because you're taking such a broad approach, you're being way less specific. So you start out with something unidentified. You've got a whole bunch of fish and you don't know what they are. So you start looking at identifying features like fins or color. In this case, when we're comparing it to this analysis method, we're looking at retention time on a chromatogram and its mass. So that's our two markers that we start out with. And as we go through the data collection process, we add more and more markers that give us a confirmed chemical identification. But you still need to purchase a standard to get that 100% confident identification. So we've got our little fisherman on the left, and he's caught 6 chemicals from his sample. It takes a lot of time and energy to identify each of these chemicals. So how do we select which chemicals we want to put our time and energy into? In this case, what's actually toxic in our sample? The approach for this is non-target prioritization. This is when you have a parallel or a separate data stream, like toxicity, that you use to select which chemicals are of interest. So in this case, someone has developed a machine learning approach to predict toxicity for a whole bunch of compounds, which is theoretically usable for things like water samples. But as I said before, we're using sediment samples. Not only is this a computationally intensive process, additional error because we're using predictive numbers, it doesn't work on messy samples. Sediment is too complicated and has too much organic matter and too much natural products to actually use a machine learning based method that the EPA tends to favor. Things like QSARs, for those of you who are familiar. But we have a whole bunch of measured toxicity data already being collected by California's monitoring programs. So using this data, is it possible to combine that data, this toxicity data, with non-targeted analysis to improve our environmental monitoring? So now that we've had our question, I want to set the stage of how do we get this data so that we can effectively prioritize our chemicals of interest. So the first thing we do is sample selection. We have historical data from the Stream Pollution Trends Program, which is my collaborator, that has values from non-toxic to toxic over a range of site types. So we used historical data and got non-toxic and highly toxic, as well as moderately toxic samples over agricultural, open, and urban locations from across California, which is the map on the left. We've also covered a bunch of geographical areas as well as a bunch of population densities. The reason we need a range of toxicities is because you can't prioritize chemicals when you think everything in them is toxic or nothing in them is toxic. So you need high concentrations as well as non-toxic concentrate or high toxicity values as well as non-toxic values so that you can properly correlate your data. So now that we've got our sample selection, we move on to our non-target methodology coverage. What I want you guys to take away from this picture is that we've used several different instrumentation techniques to cover a bunch of different chemical types. So we've got volatile and non-volatile as well as polar and non-polar compounds. So we're trying to get as big of a net as possible. So what we ended up with after casting this net is 2,449 features. This is where the prioritization starts. So collecting our actual data that we're prioritizing with, we did two kinds of tests, really three, but two parallel data collections. The first was our standard toxicity tests. This is when we take our sediment samples, stick them in a beaker, put clean water on top, and put our Hyalella azetica organisms inside, and we see if they grow or if they die. This is mimicking a natural environment. So we have chemicals that are naturally leaching out of the sediment and should hopefully mimic what would happen in a stream bed system or something similar. Parallel to that, we took our non-target extracts that were put in our instrument and we did the same kind of toxicity tests. We stuck our organisms in clean water, we stuck some of our sample inside, and we saw if they died. The reason we did this is because we wanted to confirm that we were able to extract toxic compounds. We can't guarantee without this step that we have actually extracted and analyzed and detected compounds that we know to be toxic. So these are our two, think of them as like Venn diagrams, where they line up are chemicals that we can detect and we know leach out in a natural system. So we've got our toxicity data. We've got our non-target data. Now it's time to correlate them. Hooray for all the data, lots of data points. So like I said before, this on the left is our 2,449 features. It's a lot of dots. But the point I want you guys to pay attention to are those green and blue boxes. That is those are the features, our unidentified chemicals that were correlated with toxicity. You can see all those green triangles. That is our direct, that's our extract tests. Those are extract toxicities. Those are all the features that were in our extract that were correlated with toxicity. This means our extract can extract, or method can extract toxic compounds. Fantastic job for us. Now we've got to actually do that Venn diagram step to see where these two overlap. So where are features of interest actually sitting? So on the left, we've got our growth and survival toxicity tests. We ended up with 606 features correlated with natural system toxicity. And then on the right, we've got our extract tests with 802 features that were correlated with our sample that was put, our extract that was put on our instrument and we used for detection. Together, we get 290 features. Now, what does this actually mean? Well, we started out with 2,449 features. We got it down to 290 features correlated with toxicity. That's a 10 times reduction in the amount of data we have to go through. That is a huge decrease in the amount of time I have to spend looking at, I don't know, almost 2,000 features that don't matter, or at least aren't necessarily of interest. Now, we have to check this because just because they're correlated with toxicity doesn't mean they're actually toxic. Or conversely, just because they're not correlated with toxicity doesn't mean they're not actually toxic. How effective was this approach? So I want to just talk about the gas chromatography data because that's the, I think, most straightforward of this. I know you guys are not chemists, so I'm going to walk you through this step by step. We started out with 700 features. This just means we got 700 mystery chemicals from this instrument. After we did things like blank subtraction, signal to noise ratio, basically anything that we're like, this might just be, I don't know, this is just from the instrument. This isn't important. That got us down to 573. Then we library matched. That means we took the NIST mass library and compared it to the additional data we got through our instrument process. to kind of give fingerprints to each chemical compared to the NIST library. If those two line up, we say, yes, this is probably the chemical we're looking for. Then we took those and correlated them with toxicity. So on the far left, we have any compounds, any library match features correlated with growth. Anything in bolded letters means I purchased a standard and I confirmed, yes, this is the chemical we're looking for. We've gone through the whole process and I can say with confidence, check, yes, this is absolutely identified. So some of the reasons these don't have bolds next to them is these chemicals either aren't purchasable or incredibly expensive to purchase, or like for the PAH on the top left, not found. We don't know what PAH that is. We're pretty sure it's a PAH because it's got a very distinct fingerprint, but actually doing the digging means that we have to go through several iterations of identification. But that being said, we were able to find industrial chemicals, plasticizers, 3 kinds of highly monitored pesticides, synthetic pesticide or synthetic musk, as well as crude oil components, fossil fuels, pesticide derivatives. That's DDE. I just have to confirm it. That were 100% correlated with toxicity. Things we know to be toxic, things that are of concern to the greater California monitoring program. We were able to say, yes, our procedure, our methodology is able to find toxic features effectively. Now, is it able to find everything that's toxic? Well, we've got a whole bunch of not correlated stuff that you could see, yeah, some of that's probably pretty toxic. We've got UV compounds, we've got PAHs, plasticizers, gas-related compounds, a whole bunch of mystery pesticides, or a whole range of mystery pesticides that might be there. So how do we go about incorporating additional toxicity measures so that we don't miss these compounds? One way to do this is incorporating bioassays. So we're not necessarily need to look for just mortality. We can also really hone in on what compounds are causing things like estrogen activity or aryl hydrocarbon receptor activity. So to really get an idea of what's happening on maybe a sublethal effect scale or on, to the macroorganisms you guys are interested in. By incorporating these additional levels, we're able to start highlighting different areas of that big chemical ocean that I was talking about before, and hopefully prioritize new areas of interest going forward. So in conclusion, it is possible to use toxicity results in combination with non-target analysis to improve environmental monitoring. Combining multiple toxicity pathways, that is the growth and survival versus our direct extract tests, improves feature selection and reduces the total feature space we're working with. So it narrows the amount of chemicals we're looking for. And then if we incorporate orthogonal toxicity assays like bioassays, it should increase our prioritization power with this technique. And with that, I want to thank my lovely collaborators in my lab, and I will happily take any questions. Thank you.
Thank you, Anna. We probably have time for one question, quick one.
Hi, really good job. Can you give me an example?
Yeah, just one of the ones that's on the list that was correlated with toxicity, or just like generally we found? Okay, so I believe there were quite a few terpenes and sesquiterpenes that ended up in there. I'd have to go back through and get like specific identifications for that, but there were a lot of terpenes ending up in this mix. So yeah, sorry I can't give you a better detail, but if you want to reach out to me, I will be able to give you that exact list. Thank you. Yeah.
Okay, next up, we have Luis Kumanasi, postdoc in the Sagara lab. and in the Department of Anatomy, Physiology, and Cell Biology at UC Davis. Take it away.
Okay, thank you for the introduction and thank you for having me. So today I'm gonna talk a little bit about Chinook salmon just after the salmon session. So you probably know a lot already, but I'm gonna talk about it in context of pesticide. So in California, there's 31 species of salmon and all of them are in decline, and Chinook salmon among them is no exception. In the 1800s, we counting about 2 million spawning adults, and since then it's plummeted. And last year, 2023, it was the third lowest record of adult spawning. And this decline touched the fall run, but with the winter run and the spring run characterized as endangered and threatened by the Endangered Species Act. So why is that? The reasons are complex, but can be boiled down to the use of the landscape and the water. So in the 1800s, the delta was mostly freshwater and seasonal wetland, but in 200 years, it's really shifted to urban area and agriculture. And with this shift comes the use of a lot of contaminant and pesticide. So the Bay Delta is the receiving point of urban runoff and other source of contaminant. And in addition, insecticide use is predicted to increase by 4 to 170-fold by 2099. So when we talk about insecticide, there are multiple and in mixture in the Delta, but one that I'm going to talk about today is bifenthrin, which is the most commonly detected insecticide in the Bay Delta system. And it's used for pest control, again, for example, termite or cockroach, and it's from the family of the pyrethroid. And it acts as a neurotoxin that binds to the sodium channel, affecting the release of neurotransmitter. So it overexcites the neural system, leading to the death of the pest. And it's a problem because it acts not only on those species, but also on non-targeted species in the Delta, so on all fish. And some studies have shown, for example, on juvenile Chinook, that it affects the olfactory system and them sensing predator. But also on other species endemic to the Delta, on Longfin smelt, the bifenthrin affected the size of the ashling and the yolk sac volume or the behavior. So what was a question here? We wanted to assess the effect of different concentration of bifenthrin on the Chinook that are exposed to them as they migrate the Delta. And we're interested in the aqueous concentration, but also the body residue because the pesticide accumulate in the fish throughout the diet. And it's part of a bigger project that wants to create a spectral framework or a tool that establishes a relationship between the body burden or the tissue residue and different physiology aspects. So how we did that? We had fish that were about 4 to 5 months old, and we exposed them for 10 days to different concentration of bifenthrin. So we had first a control, a low condition at 125 nanogram per liter, a medium condition at 500 nanogram per liter, and a high concentration. So we had the first exposure at 2000 nanogram per liter, but we had a lot of mortality. So we ran different exposure with lower at 1000 nanogram per liter. And I'm gonna explain why multiple exposure in a minute. But those concentrations might appear a bit higher than what is fine in the Delta. But as I mentioned, the goal was really looking into the body residue. And if we have an example for low concentration, the body residue that we end up with is close to what we have in field caught fish. So we wanted to measure a lot of different endpoints, and that's why we ran different exposures. So we needed enough fish to have strong and robust statistics. So we ran a first exposure where at the end we measure for thermal tolerance, and then the second one where we measure different endpoints as behavior and hypoxia tolerance. And a little bit more about those endpoints or physiological traits. So airport thermal tolerance and hypoxia tolerance are great indicator of the fish health as they give input about the cardiac-respiratory system. And behavior is a good indicator of fitness. I give input on the locomotion, the ability for foraging and the ability to escape predator. So for upper thermal tolerance, we slowly increase the temperature of the water and fish in which fish are until they lose equilibrium. So they turn belly up, at which we remove them from the challenge. And for hypoxia, it's similar, but we slowly decrease the oxygen. And if they turn belly up earlier, it means that they are more sensible to either the temperature or the level of oxygen. We measure also different behavior traits. So you can see it's the type of recording that we get in this video. But for that, we move fish in an arena. We let them to arbitrate for 10 minutes, after which we turn on the light and start the recording. And for the first 5 minutes, we looked into their response to the slide stimuli. And the next 10 minutes, we looked at different routine behavior traits, such as locomotion, so total distance move, velocity, cruising, the frequency of bursting, or the time they were freezing, but also trait of anxiety, chemotaxis, which is also called wall hugging, is the amount of time they spend closer to the wall, so the wall can kind of act as a shelter, or if they are more in the center of the arena. And finally, social interaction, so how much they stay close together, and we measure one body length from each other. And shrouding is a strategy against predator, as it acts as dilution or confusion against the predator. So now for the result, there's going to be a lot of results, but they are really interesting, so bear with me. Starting with thermal tolerance. So it was after the first exposure, so I don't have the highest concentration, but we can see here it's this type of graph is a survival graph, but instead of survival is the time where they turn belly up. And we have the time in x-axis and the number of fish in y-axis. And here at medium condition, fish turn faster than other conditions, so they were more sensible to the increase of temperature compared to the control. And now if we look at hypoxia tolerance, so it's the same type of graph, but this time we had no difference between the treatment. They were all similar to the control. Now looking at the response to a stimuli. So for Chinook salmon, when there is a stress, they tend to either dive down, but since the arena is really shallow, the other type of response is they tend to freeze. And that's what we can see in blue and control. And it's after, with time after the stimuli. And we group bin of 30 seconds, and we measure the time they were freezing within this bin of 30 seconds. So for control alone, just after the stimuli, they were freezing a lot, while for medium and high, they were not freezing as much, so they didn't respond this similar way as a control. And after 5 minutes, they were all back at the same level of freezing time, so we think they recover from the stress of the stimuli at that point. So Bifenthrin changed the response to the light stressor at medium and high concentration. For locomotion, there's a lot of parameters, so I choose to represent them on the spider plot. So what we have here, we have the control in blue, and if the parameter is closer to the center, it means that the parameter or the trait decrease compared to the control, but if it's outside of the plot, it increase. And if we look first to the low concentration, we can see that there's a lot more freezing than the control, while bursting over parameter were reduced. And this type of behavior can be characterized as hypoactive. They overall move less. For the medium, there were no significant difference between control and medium concentration. And for the high, it's the other way around. The freezing was reduced, while the bursting and over parameter were increased, so they were moving overall more, and we characterize this behavior by hyperactive. If we now look at chemotaxis, so as I mentioned, is a proxy for anxiety. And for the different treatment, we looked if they were more at the center of the arena or the border. And here for medium and high concentration, they tend to go a little bit more in the center of the arena. So they have a reduced chemotaxis or more like anti-anxiety-like behavior, which might suggest that they were more bold and they're likely to expose themselves to predator. For social interaction, again, we have the 4 different treatments, and this time we look into if the fish were staying closer to each other, and all of treatment were different from the control. They tend to swim further away from each other, so they are losing this social interaction. They're losing the shoaling formation, and again, shoaling is a good strategy against predators, so they might be more vulnerable to predators. So as a discussion, that's where my title is not completely true and more nuanced than that, because juvenile Chinook salmon exposed to concentration of bifenthrin show more sensitivity with temperature, but we're pretty tolerant to different with hypoxia. So I can't really conclude yet what happened at the cardiorespiratory system, and it might be partially affected, and I will need to look more into underlying mechanism. But we clearly see an effect on behavior. We saw hyperactivity combined with reduced chemotaxis, which overall suggests that the ability to hide from predator might be reduced. And it's actually have been seen in other species from the Delta, like Longfin smelt or Delta smelt. And in addition to that, they are swimming less with their conspecific, or at least more away from conspecific. So they are losing their on-predator advantage from shoaling behavior. So overall, it might, like higher concentration of bifenthrin might increase the risk of predation, which probably add pressure on this already declining population. So as future direction, we want to look more into predator cue and how predator directly affect the fish under the different concentration of pesticide. And we want to also test all those endpoints with different insecticide like fipronil, DD, or the mixture of the 3, bifenthrin, fipronil, and DD, and build the spectrum with this data. And actually with bifenthrin, the spectrum is kind of already built, and I might like this gonna present more about the spectrum just after I finish here. So with that, I wanna acknowledge all the people in my lab, the Segura lab, and Mike Leidy and his lab at SIU. And thank you, and I think I have some time for questions. I think that 2 questions there.
So it's a 2-part question. The first is, how did you administer the toxicant? And then the second is, in the environment, do you expect them to get it through the gills or through feeding?
So how we administer it is we spike the water of the tank, and then we measure both what we spike, like in the water and in the body of the fish. But in the wild, fish are both exposed to aqueous concentration and through the diet. And bifenthrin, there's a lot in the sediment as well. So it's likely that what they eat gonna be exposed in the sediment or in the water column, and then as they eat, it's gonna accumulate in their body.
Thank you.
I think I had a second question if I have time.
This one.
Okay.
Hi, wonderful talk. In your spidergram, it seemed that the low concentration resulted in one behavior and the high concentration resulted in a complete opposite behavior. I wonder if you can just comment on that, like maybe why, or if that's common.
Yeah, it actually happened sometime in toxicology with toxicology exposure. And I usually use the example of when you drink alcohol. If you drink alcohol, you might be a little bit tipsy and excited and more social at the beginning. As you increase your consumption of alcohol, you might be more sleepy. So it's kind of a dose response that's happening there. But yeah, I don't know the underlying mechanism that are explaining that.
Thank you. Okay, next up we have Jacqueline Welch, the PhD student at UC Davis, Department of Anatomy, Physiology, and Cell Biology. Her research focuses on microplastic contamination in Lake Tahoe and the effects of tire-related chemicals on threatened Delta fish species. And I'll let you introduce your talk. Go ahead.
Thank you. Hi, I'm Jackie Ling, and today I'm going to be talking to you about a project that I've been working on for the past year and a half, looking at the toxicity of 6-ppd quinone in California fish species. So up in Seattle, Washington, for the past 30 years, people have been observing coho salmon, like the one on the picture on the left, after stormwater events, where the salmon is observed surface swimming and losing equilibrium. They have increased gasping and ventilation. And then within a couple of hours of observing these fish, they almost always result in mortality. And it took a lot of researchers a long time to figure out what was happening, and they narrowed it down to a chemical that is related to tire rubber. And so they looked at the lethal concentration of this chemical in coho salmon and found that the LC50 is 0.95 micrograms per liter, which is one of the lowest LC50s that the EPA has ever seen with a chemical. They also looked at concentrations of this chemical in roadway runoff and receiving waters. And we see that in roadway runoff, the concentration is much higher than the LC50, and even in receiving waters in both Seattle and San Francisco. Sometimes the concentration exceeds the LC50 threshold, and often it's just kind of hovering right around it. And so in the San Francisco Bay Delta, it's a highly urbanized ecosystem. It's home to many threatened and endangered aquatic species. And there's a mass equivalent to approximately 43,000 whole tires flowing into the San Francisco Bay every year in the form of tire wear particles. And so 6-ppd quinone is not actually an ingredient in tire rubber, but it's the transformation product of an ingredient in rubber. The ingredient is 6-ppd. And it's a really important chemical to the safety of tires and the longevity of tires because it protects the rubber from attack by ozone, which causes cracking and crazing and can lead to blowouts on the road. And without 6-ppd, we would be burning through tires much quickly than we currently do. It is ubiquitous. It's in almost every commercial and passenger tire on the planet. And this reaction that it's designed to undergo, unfortunately, results in this unintended transformation product, 6-ppd quinone. And so this was all discovered back in 2020. It's still very recent, and California is the first to regulate 6-ppd. The Department of Toxic Substances Control listed 6-ppd as a priority product back in October. And then just a few weeks ago, the U.S. Tire Manufacturers Association submitted a stage 1 alternatives assessment, looking at a bunch of different chemicals that could potentially replace 6-ppd and scoring them based on their hazard, toxicity of the parents and the derivatives to salmonids, exposure potential, and also the performance as an anti-degradant. And this alternatives analysis resulted in 5 out of the 40 candidate chemicals, moving on to stage 2. So in about a year, we can expect an updated literature search, an in-depth evaluation of hazard and exposure potential, and also quantification of the economic impacts of potentially making a switch. But the goal of my project is to assess the lethal and sublethal effects of 6-ppd quinone to California fish species of concern. These are the 5 species I'm working with: coho salmon, steelhead, Chinook salmon, Delta smelt, and Longfin smelt. And I'm going to mostly focus on the salmon results today because I'm still working on the smelt. But for each of these species, I tested the larval and juvenile stages. And so for each species and life stage that I'm testing, I start by determining the LC50, or the lethal concentration, for 50% of the population. And we get a curve that looks like this. We have concentration on the x-axis and mortality on the y. And these curves are really helpful for risk assessments, but it was also really helpful for us because it allowed us to determine the no observed effect concentration. And that was helpful because after figuring out the LC50, we move on to sublethal effects tests. And that no observed effect concentration is the highest concentration that we use for our sublethal exposures. So Louise and Mike already talked a little bit about thermal tolerance, so I won't touch on this too much, but basically after a 96-hour exposure, the fish will be put into a bath like in the picture on the right, and the temperature heated slowly over the course of a day. And the fish are separated based on the treatment that they were exposed to. And the fish are observed until they lose their equilibrium, at which point they're removed from the heated tank and put into a recovery bath. And we chose to do this test because of those observations in the field of the increased gasping and ventilation, which suggests that there might be some cardiorespiratory effects. And this thermal tolerance test assesses overall cardiorespiratory fitness. Then we also are assessing changes to behavior. And to do this, we do a 96-hour sublethal exposure, and then we take individual fish and place them in bowls where we record their movements over the course of 30 minutes and then use EthoVision animal tracking software to track their movements. And so similar endpoints to Mike and Louise's studies, like bursting, cruising, and freezing. And we chose this endpoint as well because the observations in the field of the loss of equilibrium in surface swimming suggest that there might be some neurologic changes happening as well. And that behavior is sometimes a good test for neurologic effects. Okay, so the preliminary results for lethality are here. I have an example of one of the LC50 curves that I generated for coho salmon larva on the left. But in the chart on the right, you can see that the LC50 that I got for coho salmon larva was 0.086 micrograms per liter, which is right about what other researchers are seeing as well. So it kind of acted as a positive control for the rest of our species that we tested. The juveniles are slightly less sensitive with an LC50 of 0.138 micrograms per liter. And then Chinook salmon do not seem to be sensitive at all. We tested concentrations as high as 538 micrograms per liter, which is way higher than what we see in the environment, and we didn't see any mortality. And that's kind of interesting because there was a study that was published in BC, Canada recently, where they were testing Chinook up to a concentration of 67 micrograms per liter, and they were seeing mortality. So we think that there may be, there's obviously differences in susceptibility between species, but also potentially within species based on genetically distinct populations. And then in larval and juvenile steelhead, we got an LC50 of about 3.3 and 3.5 micrograms per liter. And this is really similar to what people are seeing in rainbow trout as well. But based on the concentrations that we're seeing in the San Francisco Bay region, we expect that 100% of coho salmon will experience mortality due to 6-ppd quinone exposure, and 14% of steelhead are expected to experience mortality as well. These are the results for the critical thermal tolerance test. We've got an example of what our data looks like on the left. This is the Chinook salmon larva. And we see that in the 100 micrograms per liter treatment, the fish did slightly better than the control. And we think the reason for that is that the fish that are exposed to sublethal 6-ppd quinone are kind of upregulating their stress response mechanisms. And so they were potentially just a little bit better equipped to handle that thermal stress compared to the controls. And so for coho salmon and steelhead, we did see changes in critical thermal tolerance at environmentally relevant concentrations. But for Chinook salmon, we did see a change, but it was at a really high concentration. This is a lot of data, so I'm not going to go too much into it, but basically the plots on the top row are for larvae and the plots on the bottom row are for juveniles. And then we have the 3 species in each column. And what we can see is there are changes to behavior, and actually every single species and life stage has a change to behavior at an environmentally relevant concentration, including the Chinook salmon, which again, we weren't seeing any mortality for. And this is concerning because changes in behavior can affect the fish's ability to escape predation and to forage for food, and can overall affect fish populations. So in conclusion, coho salmon and steelhead experience acute lethality at environmentally relevant concentrations of 6-ppd quinone. There might be differences in susceptibility between populations of the same species, like what we're seeing with our Chinook. And environmentally relevant concentrations of 6-ppd quinone cause changes to behavior in all tested species and life stages, suggesting that 6-ppd quinone may act as a neurotoxicant, which is also slightly supported with some other studies that have been coming out recently. And we've been saving our exposed fish in the -80, so we're hoping to do some future mechanistic studies, but we're in the process of looking for funding for that. And I was really bummed that yesterday I missed the poetry slam, so this is my unofficial submission in the form of a haiku. Rubber meets the road, salmon dance in toxic streams, fish drama unfolds. Thank you. I'd like to say thank you to Amelie-Segal, who's my PI, and everybody in my lab who has helped so much on this project, and all of the other PIs. The funding came from California Department of Fish and Wildlife through Prop 1. So I'm happy to take any questions.
Thanks, Jacqueline. We have time for a couple questions.
How long has this chemical been present in tires?
Since like the 50s. Yeah, so it was developed by the US military for military vehicles, and within 5 years of it being developed was in pretty much every single tire on the planet. So since the 50s, it's been around. And it is interesting that coho salmon have been extirpated from the bay for a long time. You know, can't really make any definitive conclusions about that, but it's interesting.
Thanks, great talk. What's our monitoring like for this chemical in the watershed?
The data is very sparse still. There have been a few detections in the San Francisco Bay and a couple of other tributaries really close to the bay, but in the general delta, we really don't know what the concentration is like. But we're finding 6-ppd quinone all around the planet. The highest concentration I've seen is in the Don River near Toronto. We're finding it in air and soil and urine, fish in supermarkets. It's everywhere. Thanks. Oops.
Okay, last but not least, we have Miles Daniels, assistant researcher from UC Santa Cruz and a NOAA affiliate. He will present monitoring and modeling pathogen exposure in salmon migrating to the Delta.
Great, thanks everybody. I just came from a 3-day conference. I've been talking for 3 days. My voice is like toast. I had to leave to cough a couple times. So if I start coughing during this, I apologize. I need some rest on my vocal cords. So I'm going to be talking about basically how juvenile Chinook are moving down through the system, how they're exposed to different pathogens, and what that means for their health outcomes. So I'm an epidemiologist. I think about things in this kind of epi-triad, right? We have a host, salmon, we've got some pathogens out there, and the environment. And what we're really trying to do is figure out how all those things are interacting, right? It's not just about the concentration of the pathogens. It's not just about the susceptibility of the Chinook or the temperature they're moving through, but it's all the combination of those three things, how that really plays out. And so this goal, the goal of this project is really to fill data gaps, essentially, around disease transmission in Central Valley Chinook salmon. And we're kind of doing it through a 2-tiered approach. We're taking environmental monitoring data as one to see what's out there, and then merge that with kind of models to try to understand what can we predict to occur under these different circumstances. So my goal for today is really to kind of give you a brief overview of this project. I should have mentioned this was a project funded by Prop 1 with some other funders, which I'll get to at the end. And so there was a lot that was done. There's been a few publications coming out, more to be expected, but I really just want to pique people's interest because this project was a 2-year project. We discovered some things, but we really developed more questions than anything. So here are the 4 main objectives. Step 1 is really just screening what's in the environment. There's been some work done, but it really hasn't been done in a systematic fashion. So we need to know what's in the environment, right? Before we can kind of start looking at effects on salmon. If we were to find no pathogens, it would be unlikely that any fish would be getting sick. It's unlikely to find no pathogens, of course, but that's a good thing to first do. Of course, if we find pathogens in the water, that doesn't really mean that all fish are getting sick, right? There's just different susceptibilities. And so we want to be able to look at what are fish actually experiencing. And then we want to build a model to simulate this, right? I'm a modeler. I like taking data, putting it in a model, playing around with things. And then we want to build a web-based decision support tool to share this information. I'm not going to really talk about that today. Really, it's more 1, 2, 3. So screening the environment. What I mean by this is really collecting water samples, right? Fish are moving through water, of course. We want to collect water samples to see what's in there. This map off to the side over here is showing you where our 12 sites were. We basically had 10 sites on the Sacramento River, 2 sites on the Feather River, kind of longitudinally spread throughout those systems. And we'd go every 2 weeks, grab water samples for 2 years and have those analyzed. We analyzed a few different things. One is if you've heard anything about diseases in Pacific Northwest salmon, you've heard of sea Shasta. It's very prevalent. It's endemic. It's supposed to be here. They've evolved together, salmon and sea Shasta. But we wanted to really identify that pathogen a little bit more and look into it because there's been some evidence indicating that it might be having some population health impacts. But we also wanted to be more broad too. We wanted to say, well, what else is out there? Guaranteed there's more things than sea Shasta out in the system. It would be strange if there wasn't. So we did a kind of a large scale, kind of broad screening approach using some assays developed by Christy Miller up in Canada to look for 36 different pathogens that are known to infect salmonids. This is kind of a mix of bacterial, parasitic, as well as some viruses. So that was screening the water. But as I mentioned, it's not just about what's in the water, right? We need kind of multiple lines of evidence here to say that, yep, we see it in the water. Do we also see it infecting fish? So to get at that a little bit more, we did what are called sentinel fish trials. This is essentially where we take fish of a known infection status, put them in enclosures in the river, let them be exposed to ambient conditions, and then kind of monitor them at different time points and essentially sample their tissues, which we think are targeted by certain parasites to see if we find, yes, this parasite, which targets the intestine, is actually in the intestine, i.e. there is some infection occurring. And we did this while screening the same collecting water, et cetera, so we can have that kind of matching up of evidence. And we did this at 4 sites shown on this map here. So Red Bluff, Wilkins Slough, so kind of an upper and a lower on the river, as well as Gridley and Boyd's Landing on the Feather River. So that's screening tissues, but many of you are probably wondering, well, you put a fish in a cage at one location. There are all kinds of cage effects that occur with study designs right around that. And so natural fish are moving through their environment in a much different way than a fish stationary at one location. So to try and get at that a little bit, we also partnered with folks at U.S. Fish and Wildlife Service up at Red Bluff and took some of their rotary screw trap samples and looked at what kind of wild outmigrating salmon are doing. For these fish, we only looked at their gills. So a gill is kind of a good indicator, but not necessarily going to tell you if it's actually infected. So some of those samples are also assessed via histology with our collaborator, Dr. Scott Foote, to kind of really be able to say, yes, we see the pathogen and we see the effect in the tissues, kind of the gold standard. The model, I'm not going to go into it in depth. If you want to talk about it, I would love to talk to you more about it. But it's a pretty simple model. It's used for not just salmon pathogens, but any other type of infectious disease, essentially. It's an SIR model where you break up your population into a susceptible, infected, or removed or recovered group. And then you can kind of really look at what the population dynamics are in response to a pathogen exposure. For this model, we're really, again, focusing on sea Shasta. It's a well-documented pathogen. There's lots of other literature that's been done and lots of other studies that have been done looking at like LC50 curves and things like that. So we can kind of leverage that when we're modeling this and use that kind of out of basin information. But I will note that this is a flexible approach, right? If you have a pathogen of interest, the framework is there. It's really a data limitation thing. If you don't have the data to be able to parameterize your model, it's not going to work, but the framework is very flexible. Okay, some results. This was a large effort. There is a lot more than I can talk about in this short amount of time. I'm going to give you a very high level overview. But again, the goal is to pique interest, and we're always interested in having more collaborators and involvement. So if this kind of relates to your work somehow, reach out. So what did we find in the water? Very busy map. There's a lot going on here. This is really just focused on sea Shasta and what we observed in the water. So you've got the map. You're generally familiar with that, I'm pretty sure. If you look at that pie chart at the very top, saying global prevalence, that's just saying out of all the samples we took, how many had sea Shasta at a detectable level? And it's about 3/4. So it's pretty prevalent in the environment. Next, you can look at the kind of circles along all those little locations that we sampled, right? The larger the circle, generally the larger the levels of C. shasta we detected, right? And so this is kind of interesting because you can see there's kind of a clear spatial pattern occurring, right? There's kind of hot spots that we see in terms of where we see C. shasta on the systems. And that could be pretty important for delving deeper into what's happening in terms of population responses, salmon exposure, et cetera. And then those little bars, a lot of information here, those little bars are essentially giving you the temporal signal. So each little grid area represents a sample from a biweekly sample, color-coded to that scale at the very top there. So red, high levels of concentration, blue, very low levels of concentration. And you can see there's not just, right, spatial variability, but there's often lots of temporal variability as well. And so when we're talking about Chinook runs, 4 different runs, right, moving through the system at different times, different life stages, this is going to play out pretty interestingly in some of the modeling we do in terms of exposure and risk. So that's C. shasta, the real pathogen that we kind of focused on. But again, we looked for other things as well. I don't expect you to read this, although these screens are so massive that you actually can, which is great. So we've got basically the 36 pathogens we screened for. They're scaled to just relative abundance from the molecular work. It's not like there's more of them. It's just kind of a proxy for that. But what I want you to really focus on are the top ones, right? We found about 6 or 7 that were frequently detected in all of our sites, for the most part, at pretty high levels. And so this is just begging the point that, yes, you know, C. shasta is out there. People know about it. It gets a lot of press. There's other things that are going on. You know, C. shasta is not working in a vacuum and affecting these fish. There's other pathogens that might be interacting with that. So it's good to kind of keep that in the back of our minds when we're looking at diseases and how they're affecting populations. That was what we found in the water. What did we find in some of the fish? This was some work published in Conservation Physiology, so I don't have time to go into it in detail, but read that, talk to me, and we'll discuss it more. Basically what we did, I would say focus just on that top left. Yeah, your left, C. shasta plot, it says. There's 2 kind of sections to this plot. There's a gill and a kidney tissue. You might be able to see it. I'm not gonna be able to find the cursor. Let's see, maybe I can. Laser pen. So that's day 0. This is day 7. This is day 14, day 21 after exposure from being put in the river, right? The bars are representing essentially the prevalence, just was it detected or not. And then these little cluster of dots is representing kind of the log abundance in terms of gene copies. What we see is that basically by day 7, all the fish have it detected on their gills. That's not too surprising, right? I mean, the gills are basically a filter. They're filtering all this water. So if we find it in the water, it's likely to be found on the gill. What's more interesting is we do find it in the kidney tissue at day 14 and day 21, which meaning maybe it's going systemic, and it is actually a level of infection that we care about. 5 minutes, all right, I gotta hurry up. So we find it in the water and we find it in the tissues. Another thing we did, which I didn't really go into too deeply, was gene expression. This is just looking at different genes and how they're upregulated. So we looked at pro-inflammatory, anti-inflammatory, stress response. This is in the paper. I definitely don't have time to go into it, but there are associations between exposure and the upregulation or downregulation of certain genes. And that's just another line of evidence saying, yes, these fish are infected and physiologically responding to this infection. And that can have important implications, right? For avoiding predators, eating food, energy, the whole list of things. This is what we found in the wild salmon. So all those other fish we were looking at, right? Those are kind of caged fish. This is the wild salmon. So this is about 2 years of sampling, 80 fish per year at Red Bluff Diversion Dam. All the pathogens that we screen for are on here. Don't really pay attention to that so much. More just focus on that we do see C. shasta very frequently in our samples. And so again, this is just another line of evidence that we see it in the water, we see it in the caged fish, and we see it in the wild fish too. And we're seeing it in the wild fish that have made it to that point in time. All right, integrating the data into a disease transmission model. So this is not going to go, I'm not going to be able to talk about this very detailed, but I will kind of give you a very high level summary. And again, reach out if you want to talk about this more, if you have ideas on how to improve it or collaborate, we're all ears. We kind of broke it out into these 4 main components here. So we need to know when fish are moving down through the system. That's our movement model. We need to know what they're exposed to as they're moving downstream. So we have a variety of models to do that. We care about temperature. We have a temperature model. We care about hydraulic conditions. We can have a model for that. And we also have kind of a sea Shasta model based off some empirical data. So we can get basically a time series of their exposure. We take that information and then we assess risk. And we do it through this approach called QMRA, Quantitative Microbial Risk Assessment. This is very popular in human health, where I used to work a lot in. It's used by WHO, EPA. It's the way that you assess risk, right? Say if you're swimming in a contaminated river, et cetera. So we're just applying those same principles to fish. And then I already mentioned this, where we kind of break it out into this SIR model, this large kind of compartment model. We combine all that to be able to say what fraction of the population is susceptible, infected, or removed from being exposed. Just like some other dose response people over here, we want to have a dose response curve, right? We assume there is a dose response relationship. The higher the dose, the greater the response. This is coming out in a paper hopefully soon, where we basically took 4 parameters. We have sea Shasta concentration, that's very relevant, water temperature and channel velocity and duration of exposure. Combine those kind of in a mixed effects logistic regression approach, do some Bayesian fitting, and we want to be able to predict what's the infection probability given your exposure history. Here are those results from those caging studies I talked about. So Red Bluff at the top, Wilkins-Sioux at the bottom, Gridley at the top, Feather River, and Boyd's Landing at the bottom of Feather River. The grayish is the prediction from the model, and the colored one is like the actual empirical data that we got. So you can see that one, the model fits the data pretty well. R squared is greater than 85%, so that's good. But more interesting to me, aside from a well-fitting model, is that there's some pretty interesting dynamics again occurring here that I kind of touched on already, where for the most part, after day 14, which it might be hard for you to see, but prevalence of infection is very high. And this is in the intestine, I should note. So this is actual infection of fish. They're actually infected with sea Shasta. It's pretty evident there. But there's a lot of variation in day 7. Some day 7 fish are not, and some day 7 fish are. And so there's a lot of variability there. And we also have to take this in context, right? You know, some fish might be moving through the system very fast and spending less than 7 days in the system. Some might be spending a little bit longer in the system, such as natural reared fry. And so how this plays out in terms of population dynamics is something we're exploring a little bit further. All right, wrapping it up, conclusions. We wanted multiple lines of evidence, right? Not just a single line of evidence. So we have water, sentinel studies, rotary screw traps, and they really do point to sea Shasta being detected pretty frequently in our samples, as well as other pathogens of interest. But it's not just that. We see it in the gene expression as well, in the histology, that these fish are responding to these infections. So it's important for them. But this last point is very important and something we really want to dive more into. We really need to understand the fate of these infected fish, right? Do they recover? Do they not recover? Are there side effects, right, that occur, allows them to be eaten more frequently or just consume less energy? All these other things that we really need to take into account. We've kind of scratched the surface, but we need to do more. I think my time is up, so I will not go into future efforts, but I do want to say this was a large effort. A lot of people here involved on this, and a lot of names that aren't even on here. This was a big effort. I'm just here talking about it. Funding again from CDFW, Metropolitan Water District, as well as some from California Department of Water Resources. And I think I put those up at the very beginning of the slides. I want to give them credit, and I'll say thank you.
Thanks so much, Miles. I think in the interest of time, we're going to move on to the early career awards. Thanks again for your talk.
All right, thanks everyone. I have some good news and some bad news. The good news is we're going to finish early. The bad news is because I haven't finished calculating all the scores for the early career awards. It's basically a function of the fact that we had a really great crew of early career scientists at the meeting this year. And we also had a lot of judges volunteer to spend part of their time at the meeting going and meeting with the early career scientists, listening to their talks and taking detailed notes to evaluate which would be our winning presentation. So keep your eye on your inbox. We'll be announcing the winners over email. I did want to share a couple of things with you, for a little background about the process. All of the early career judges are scientists. Some of them are sitting in this room. And they spend a lot of time looking at the posters. I think a lot of us who, are maybe mid-career, later career scientists, we kind of honed our chops in meetings like this. Even if you're new to the system, if this is your first IEP, these are the types of meetings where when you are a grad student or a scientific aide or starting out as a, entry-level environmental scientist or biologist, these are the types of meetings where you hone those chops. You learn how to give a poster, you learn how to talk to experts, you learn how to talk to non-experts. And, I think a lot of people maybe are a little scared of doing that. But, I think the reality is this is an incredibly welcoming environment for early career researchers. This is an excellent place to get started because you're surrounded by a lot of folks who've been doing this for a long time and want you to succeed, and they want you to be better, and they want you to evolve as a scientist. So I just wanted to highlight, because I, can't give up a 20-minute speaking slot. Geez, when am I just going to sit down? No, I wanted to highlight some of the words of encouragement that our judges gave to these next generation of IEP scientists. So there were some really excited judges. Wow, that is a complicated system of data, but nice job pulling together a whole lot of it. Lots of use of the word cool use of existing monitoring to ask questions, and a cool pilot study to address a clear monitoring gap. Important work with an exciting future, very important research with huge implications. The presenter was very articulate and knowledgeable about her topic. Super interesting, well-executed study with extremely important conservation implications. Also some tips on, people weren't afraid to pull punches. How would someone who uses this data explain it to their mom? Now, notwithstanding, there are a lot of moms in this room with PhDs in ecology. So you know what? I think they're going to be doing a lot of explaining to us about this data. But still, good to think about. How would you explain this to perhaps not all our moms have PhDs in ecology or biology. How would you explain it to someone who's not a scientist? How would you explain it to someone who is a scientist who's not in your expert? There's a huge range of expertise, as we've seen over the last 3 days. And being able to create a compelling and informative IEP talk is going to depend on your ability to speak to not just those people in your niche, but everyone in this whole auditorium and listening at home. For example, one of the judges, a nice addition of plain language to your talking points that really helps the broader audience's understanding. So anyway, that's it for me. Keep your eye on your inbox, and we'll be announcing the winners soon.
Yes, thank you, Ted. Thank you, everyone. I've had a lot of people come up to me over the past 3 days and, oh, Rosie, you did a really nice job, like putting together a great workshop. Like, what did I do? I took an amazing group of submitted abstracts and I ordered them in a way that seemed to kind of make sense with the help of our whole planning committee. The great part of this workshop is the caliber of the science done at IEP and the caliber of the presentations put together from that science. So I think everyone deserves a huge round of applause. But I do want to give a special round of applause to all of the people who really did the behind the scenes work, getting the building reserved, getting the audiovisual equipment working, getting the coffee bar in place, doing mic running. So can I have a huge round of applause for the planning committee and our IT folks in the back? And one more round of applause, in particular for the IEP program support team, Christine Job and Stephanie Fong. Christine, in the back. And with that, I think we can wrap up the 2024 IEP workshop. Continue doing the great science you do and look for next year's workshop planning to kick up in the fall. So if you enjoyed the workshop and want to participate more actively next year, we'll be putting out a call for planning committee members in the fall. Yeah. Anything I miss? All right. Well, thanks again for coming, and all of the recorded talks will be posted online in a couple of weeks once we finish all of the closed captioning. All right. Have a good night, everyone.